Raz, dwa. Słychać.
Tak wysoko nas posadzono.
Once we have an idea, we just do it together. So we can control that.
Dobry. Jeszcze raz, tak?
Ladies and gentlemen, good afternoon. I have a great pleasure and great honor to welcome you at our Basic Sciences session at International Medical Students Conference, this year's edition. And at first, I wanted to say that we have a very honorable patronages this year. As you can see, there is a Polish Anatomical Society, Polish Physiological Society, also Polish Biochemical Society, and Polish Society for Histochemistry and Cytochemistry. And one of the most important things, so we have a really exquisite scientific committee. And please welcome with me Professor Piotr Leidler, Professor Tomasz Brzozowski, Professor Jerzy Walocha, Professor Marek Stanak, Professor Adamek unfortunately will be absent, and Associate Professor Krzysztof Gil, and Dr. Mariusz Gajda. And my name is Kinga Michalewska. I am the session coordinator. Here is my coordinator, Maria Koshi. Please welcome her as well. And she will be leading the second part, which will be electronic posters. And I just wanted to shortly remind you the most important rules. So, the official language of the conference is English. The decisions of the jury are final and unquestionable, and they will be announced just a short time after the end of our session. It will be like 15 or 20 minutes, I hope. And another two important rules are the timing. So for oral presentations, it is 7 minutes for presentation and 3 minutes of discussion. And for poster presentation, it's 4 minutes for presentation and 2 minutes of the discussion. And I hope that each of you will be looking at the timer. It will be placed in a minute in the corner of the screen, because with the 20 works, we really have to look at the time and try to be exact about this. And first, second, and third place will be awarded, as well as 2 honorable mentions, so 5 prizes for our sessions. And prizes are awarded together for both oral and poster presentations. Okay, so now it's time to place the timer. And please, the first participant, so Ilya Dmitrievs, with the work Association of the PSMA6 Promoter Poly-A, Polymorphism, with type 1 diabetes mellitus in Latvians.
Good afternoon, dear audience, dear commission.
You can wait now for your work is here. And just give me a minute to place the timer.
One more time. Good afternoon. My name is microphone. Okay. It will be better. Good afternoon. My name is Ilya Dmitrievs. I am from the University of Latvia. Don't want to work.
Yeah, it should work like this. It didn't work. Nie działa.
Yeah. Thank you. And I will begin. So, proteasome is multicatalytic protease complex, which is play a critical role in degradation in intracellular proteins, and you see that PSMA6 gene codes the protein which is called alpha-1, which is very important for the forming of the 20S proteasome core structure. This one. Oh no, he don't want to work. No. Oh my God. Okay. No. Next. I apologize for this. So, why poly-A is so interesting for us? Because it's important for the transcriptional regulation, for recombination, for the spread of histone post-translational modification, and a lot of another reasons. Here you see the full motif of our microsatellite, but we analyzed only this variable part. Aim of our study was to find the probable association between type 1 diabetes mellitus and poly-A tract variation in the PSMA6 gene promoter in the 14 human chromosome. We use case control study where was 147 cases and 323 healthy controls. We used DNase extraction with the DNA purification kit, electrophoresis, DNA sequencing, and fragment analysis for the determination of the microsatellite fragment length. Here were observed a lot of genotypes, but for us it's interesting only for, as you see, the genotype 215.16 and 215.17 is appear to be for type 1 diabetes mellitus protective, but these genotypes 216.17 and 217.17 appear to be association with the type 1 diabetes mellitus in the Latvians. Were observed 6 alleles. As you see, allele A18 and A21 appear to be protective in Latvian population, and allele A20 appear to be associated with type 1 diabetes mellitus. And here you see we made analysis with Genomatics program, and you see that numbers of transcriptional factors change. Here 8 transcriptional factors, here is 10 transcriptional factors, and this is POLEA change the number of these transcriptional factors. How you see, POLEA tract doesn't change the DNA structure of this studied locus. And also POLEA tract does not change the structure of RNA of this current locus. And conclusion that genotypes A1920 and A2020 were significantly more frequent in cases and appear to be associated with type 1 diabetes mellitus. And genotypes A1819 and A8020 were less frequent in cases and appear to be protective in case of type 1 diabetes mellitus. And POLEA tract length do changes number of bone transcriptional factors, and POLEA tract doesn't change the DNA and RNA structure of studied locus. And thank you for your time.
Is there any questions from our jurors? Please, please.
Okay, thank you. My question is about this variant, this microsatellite repeat you studied, because you also showed that there is an allele with a transversion from A to C. So you analyzed only number of repeated adenine residues, or do you have also results on frequency of this A to C transversion?
Thank you for the question. No, we don't have results of this small SNP because we analyzed only this variable part in the microsatellite begin, as I showed in the previous slide, yes, and we don't have any data because in our earlier findings in Latvian population, we became to know that this is part, they do not change as we want to know, if we want to analyze this, and it was not interesting data for us. We analyzed only this POLEA tract, not this SNP.
This STR results are very appealing and associations are strong. However, from genetic point of view, it's rarely observed that you have effect in heterozygous individuals.
Yes.
And if you appoint these alleles like risk 1 A19 and protective A21, you should observe stronger contrast between homozygous individuals, not heterozygous. But homozygosity was with the same frequency for both groups, for controls and diabetes. That's my question. Perhaps one of these alleles is carrying this A to C.
Unfortunately, now I can't answer for this question because I don't interested with this SNP and we don't make any correlation with the POLEA tract, this homozygous and this SNP.
Okay, thank you very much.
Interesting another because I made analysis with adiposity and bronchial asthma, this POLEA tract and this protective to A21 allele. And this allele is figured in every 3 pathologies and it is protective and it's statistically significant that this allele is protective in 3 pathologies: for bronchial asthma, for type 1, and for adiposity. And it was an interesting result. But
I mean, obesity, not that.
yeah, no, I'm
Okay,
sorry,
thank you.
obesity, yes.
Thank you very much.
So is there any other questions? So thank you for your questions and
Okay. Oh, I'm sorry. And how will I change the slides?
So
The left one or the right one?
it should work like this. So right one, left one,
Okay.
and here you have laser.
Mhm.
So you can stay here or here as you prefer. If you will stay here with the microphones, if you don't want the microphones, you can just
If I just talk really loud, will everyone be able to hear me? Okay. I'll just forget about that then. Do I need the microphone? No? I do? Okay. Hello? Okay. Yes. So my name is Marcin Kuzma, and I am a second-year student here at Jagiellonian. I study as part of the English language program. And I'm going to describe to you today my research project that I did this past summer with the Indiana University School of Medicine Northwest from back home in the States where I'm from. And so the title of the abstract I submitted is Sensitivity of E. coli mutant strains to peptidoglycan recognition protein-induced killing. And before we get into the research, it's important for me to kind of describe to you a little bit about peptidoglycan recognition proteins. And so what these are bactericidal innate immunity proteins. They're bactericidal to both gram-positive and gram-negative organisms. And importantly, they're bactericidal to antibiotic resistance bacteria, for example, MRSA. So this is where we can have implications for the future by combating the problem that we have with antibiotic resistance. Specifically, these proteins recognize bacterial peptidoglycan, as the name suggests, and the genes for these proteins are conserved from insects to mammals, so we see them across species. And there's 4 types in humans, and they've just been named 1 through 4. And so the team at Indiana University that I was working with, they've basically been working with PGRPs or peptidoglycan recognition proteins since their inception about over a decade ago. This has kind of been the focus of their work. And here is just a model that they've composed so far of the mechanisms they've outlined about PGRPs. And kind of the main takeaway from this diagram for now is right here that these PGRPs induce envelope stress via oxidative stress, thiol, and metal stress. And that's going to result in membrane depolarization, inhibition of the ATP synthase, energy depletion, and then inhibition of peptidoglycan synthesis, DNA, RNA, and protein synthesis. And ultimately, that's going to result in your bacterial death. And so the focus of my research during my time over the summer was to look at the effects of PGRPs on a mutant collection of E. coli, the Keogh collection. And so this collection has certain genes, each one a unique gene, knocked out. And we wanted to analyze these mutants to kind of see the role that PGRPs play on some of these genes and which ones may be more sensitive to killing. Essentially, this was an attempt to further outline this mechanism of the action of peptidoglycan recognition proteins. So before we could start the project, we needed enough PGRPs to complete the work. And this is a very tedious process. Before I even got there during the summer, progressively each day, they work on purifying some of this protein. Around the facility people, or the people working on this project, kind of consider it as precious as gold because it's very tedious to purify this protein. And so basically the steps that are outlined, you need to prepare a buffer, prepare the sample through dialysis, then purify the PGRPs through elution, and then you store this protein at about -80°C. And just to give you a little, to quantify it a little bit, you get about per sample, or per soup as we like to call it, you get about 500 micrograms of this protein. And for each sample that we inject, say a pool of E. coli, we use 100 micrograms. So it takes a very long time to kind of make this purified protein. And so once we had enough of this protein, we were able to start the research project. And what we did was we took pools of about 10 to 12 mutants and treated them with about 100 micrograms per milliliter of the PGRPs. And once we did that, we let them kind of harvest for 3 hours, and then we diluted the samples to kind of standardize everything into these 96 well plates. After that, we kind of left them in a spectrophotometer overnight and measured the optical density every 10 minutes. We looked at the growth curves for these samples and kind of identified which ones had accelerated growth curves in respect to each other and in respect to the wild type. And so from this, mutant candidates were selected, the pools, and then we took those pools and then individually assayed the mutants from there to look at the individual samples. And so the results of the screening were very promising. We deduced that there was greater sensitivity to PGRP killing in E. coli mutants that specifically had many mutants in energy-generating metabolic steps. And so further assays of the individual mutants from these pools also confirmed this, and that there was essentially greater resistance than wild type to the PGRP-induced killing. And so here we have a table of kind of the results of the main genes that we saw had greater resistance. And kind of the main takeaways from this are that you do get this increase in resistance, and this is the fold increase, so the just multiples of 1, 14 as much, 48 as much. And the second thing is that these genes are all essentially in energy generating metabolic steps, such as the citric acid cycle and the electron transport chain. So the implications, we showed that certain genes play a greater role in the sensitivity to PGRP-induced killing. And we outlined another mechanism kind of to add to that, that diagram I showed earlier in the presentation. And that, that mechanism is metabolic stress. And so that's another additional component that we kind of discovered through this project. And for the future, we kind of want to look at how is this metabolic stress generated exactly, outline the mechanism, and then also how it relates to those 3 original mechanisms that have been explained, which were oxidative thiol and metal stress. And so ultimately the goal through working with these PGRPs is to hopefully at some point come up with mimetics that work similar to peptidoglycan recognition proteins that can be possibly used as pharmaceuticals. And just acknowledgements of the team that I worked with at IUN this summer. And just to thank Indiana University for allowing me to be there. And the next slide should be questions. So now I'll take any questions you may have. Actually, I'll probably keep this.
Okay, so
Sorry?
I invite you here and we'll give this microphone. Are there any questions from the jurors?
Yes. Yes.
I would like you to explain more about the methodology. That means you added to the pool of the mutant cells this purified recombinant protein, but why you monitored for accelerated curve of growth of bacteria, not decelerated. Killing of bacteria would cause slower increase in absorbance.
Exactly.
Yeah.
That's a really good question. So maybe, okay, I can't go back to the diagram, but what we were looking for was sensitivity of these mutants to PGRP-induced killing. So what we were looking for is, okay. So what we were looking, here we outlined the resistance of the mutants to PGRPs. And so what this means is that these E. coli mutants were more resistant because they had the gene knocked out. So what that tells us is that that gene is important in PGRP-induced killing because when they don't have this gene, they're not very sensitive to death. So that's kind of the way we looked at it. So you had to look at an accelerated growth curve because that means there's more resistance to PGRP-induced killing.
Now it's clear to me, but another question is raised by this explanation. Are this collection of E. coli real strains from patients? Does it mean there exists some resistant E. coli on the large, which precludes what you just said, that the peptide can be used against some resistant bacteria?
Well, what I'm referring to here is that, you know, this library of E. coli, they just have particular genes knocked out.
So it is artificial library, yeah? From
Yes,
mutagenesis.
yeah, it's the Keogh collection. Because
Thank
what
you.
we are looking for is we just want to outline the specific mechanism of the action of PGRPs as it relates to E. coli.
All right, I didn't know Mr. Keogh, but I don't know where this acronym or proper name comes from, but this is artificial library
Yes,
done
yes,
by
yes.
knockouts of
Yes.
bacteria. Okay, thank you.
Are there any other questions? If not, that thank you very much for questions and thank you very much for your presentation.
Thank you.
And just to clarify for all the other presentations, there is a possibility also to present from here using these microphones or from here using this mobile one, or even the one which you have to attach to your trousers if you have trousers today on you. So, so there are 3 possibilities and even 4th, because you can also sit here if you prefer to.
Good afternoon. My name is Patricia Donat, and I would like to tell you something about computer application control on the basis of bioelectrical signals. Włączysz mi proszę prezentację? Newton. Our project is dedicated to paralyzed people who are conscious but not able to move. Despite the fact that disabled people constitute a large percentage of our population, most electrical devices are not adapted to their needs and capabilities. It causes difficulties in communication with others and results in their isolation and loneliness. Of course, devices designed to help paralyzed people are available on the market, but realistically, they are very expensive and very difficult to get for ordinary people. The aim of our project was to design an interface that allows disabled people to communicate with others. Did you know that even in deep paralysis, the mobility of at least one of eyeballs is retained? That's why we built a prototype of device that allows you to control cursor using eye movements. We took into consideration specificity of disabled users and individuality of every human. Devices operations can be divided into several steps. First, the EOG signals are acquired using a specialized measured module. Then the microcontroller generates 4 digital signals in response to the recorded 2 analog ones. And then BALMOUS generates responses, which allows us to gain control over cursor. Additionally, we created an application that facilitates further usage of the interface. In EOG, we use the fact that eye is an electric dipole, which allows us to measure differential potentials generated during eye movement. It is worth mentioning that EOG is an non-invasive method. We measured vertical and horizontal signals, so we needed as many as 5 electrodes. The results has a small amplitude, and there are some interferences caused by blinking, but quality of measured signals were actually really good. In the picture, there's a part of prototype of the device we used during tests. In the final version, it will be, of course, much smaller. The device allows to move cursor in directions up, down, and right, left. We didn't use eye tracking. The cursor moves to a predetermined distance when a trigger value of signal is detected. This improves the stability of devices' operations. The LabVIEW application was written for paralyzed people to enable them to communicate with caregivers. For this reason, there are 3 basic actions. The text panel allows to write a message. Search panel looks similar to text panel, but it draws carer's attention on need to find something, to bring something for patient, for example, medicine or blanket. And the phone panel allows to express the desire to contact somebody, for example, call a doctor or a nurse. At this stage, the device allows only to move the cursor, so the application doesn't require pressing any key. If the cursor stays on the button for 3 seconds, the application passes to next panel automatically. To avoid excessive accumulation of characters on one panel, first you have to select a group of characters, which results in displaying them on the right side, and then you can select a specific character you want. Phone panel allows to choose a method of selecting contact person. Sub-panel keyboard allows to type a phone number, which can be helpful for people who want to contact somebody for the first time. And sub-panel contact list allows to select a person from the list of previously entered people. Our device not only allows paralyzed people to communicate with others, it can be used in many other ways. For example, in biofeedback. Biofeedback is a medical method of increasing the possibilities of mind through training involving performing actions, mostly playing games, without using keyboard or mouse. You can only use eye movements, or in another version, EEG signals. These workouts help improve concentration, memory, speed of learning, and improve mood or teach relaxation. And they are recommended both in serious diseases, such as autism or cerebral palsy, and for healthy people who want to improve concentration or whose work is associated with excessive stress and highly responsibility. To sum up, the interface provides a non-invasive method to communicate with paralyzed people. We have some plans for improvements, including solving linking problem and making the device smaller and wireless. But our prototype already has many advantages and potential to help disabled people to connect with others and to prevent their isolation. Thank you for your attention. If you have any questions, you can ask me now.
Are there any questions from the jury or from our audience? Okay, there is a question from a jury.
Oh, congratulations. It was a unique paper because you talk about medicine using technical device. And my question is about the, actually I do have two questions. First, the shape of these potentials. You think it is kind of this EOC or AOG, right? Are they spike potential rather, or it is just rising and decreasing curve type? I'm trying to imagine how it looks like under normal condition and try to compare that.
Well, actually, they are way bigger than, for example, in EEG, in electroencephalography.
Mm-hmm.
So it's easier to measure them. Of course, they're a little bit smaller than in ECG, in electrocardiography. But their amplitudes are enough to measure them and to even change the threshold of that is.
So they were not, I would say, fully normal, right? Like according to one picture you showed me. But again, I've seen depolarization kind and
yes, because
repolarization.
Here on this picture, yes?
Yes, exactly.
So this shows what it looks like when you look straight forward, and then when you look, for example, up and down. And it's actually...
So it is like eye movement, right?
Yes,
up and
yes,
down.
yes.
Only
It's
without...
up and down, and again forward. And there is actually about 1 volt when it's amplified. So it's enough, really enough to measure them.
And you said that practical implication coming out from this study is that these disabled people can communicate, right? Or they can just, they are aware but disabled. And my point is, you practiced this in normal volunteers, or it was just in
well, actually,
perhaps stroke patients?
Well,
or,
actually,
no, probably
it's
not.
a prototype, really prototype. So we use only me
Our
and my
cells.
colleague use,
Yes.
use it, but it's able to, using only eye movements, to write something using this application. Because when we used our eye, moved our eyes, then the cursor moves on the screen and you can type these few letters. I know it's slow for the moment, but for somebody who haven't any other contact to express their emotions, opinion, and so on, it's actually really helpful, I think, because, oh, the time is over. So I think it could be really helpful for people with paralysis or other disease.
Disabled people, right?
Yeah, yes.
Thank you very much.
Did you try to write a word that way? How long time it took?
It depends on how long
I
the
know,
word
but
is.
just
And how fast the cursor moves, because we can change it too.
No, but then,
for example, to simple sentence, a minute, it's enough, yes.
so, okay, the idea is interesting, but it requires improvement, certainly, but it's a great idea.
Yeah, it needs some improvements, we know that, but it's
Yeah, but that's
actually...
obvious.
Okay, so thank you very much for the discussion and for your presentation. Please let me give you a certificate. And now there will be a presentation, Regulation of Restriction Factor Expression in HIV-1 Vertically Infected Children by Martin Borslik. And I also wanted to add that I'm very sorry for these problems. It's just because there is a small interference of our timer and our presentations, and I'm really sorry for all the additional stress we've caused by it.
Ladies and gentlemen, good evening. I'm a Czech military medical student from Hradec Králové, and I've done this study at the George Washington University in Washington, D.C. And in the next few minutes, I will tell you a short insight into pathogenesis of HIV in children. But first things first, 2 Americans get Ebola and everybody wants to wear a biohazard suit. Millions of people live with HIV and yet no one wants to wear a condom. I think this is really alarming and we should think of it because 70 million people have been infected with HIV so far and 1.5 million die every year. HIV isn't only a problem of Africa, it's everywhere. I've been working with perinatal infected children, and even though these guys represent only 4% of HIV infected population, they stand for 20% of death, which is alarming and horrible. If you have a look at the curve of viral load, so when you get infected, the virus goes up. And since it causes death of cells, of CD4 lymphocytes, which represent the main group of specific immunity, these drop here. But since people are generally really hard to kill, so we've developed some kind of mechanism that will control the virus, at least for a little while. So people, after infection, they live without any treatment for usually 8 years. But we have 2 groups. The first of them are repeat progressors that are characterized by allele HLA-B35. These people live for a shorter period of time, which is usually from 1 to 6 years. On the other hand, we have slow progressors characterized by HLA-B57 haplotype, and these people, they live for decades, basically, with little or no symptoms at all. So this goes hand in hand with HIV antigen in plasma and also the production of antibodies. We test for HIV usually after 1 month of infection, and then we repeat it after 3 months. So as of now, we usually have some factors that tell us how the disease is bad. It's CD4 count and RNA values, which is copies of the virus, but it's not very good for us to tell how it will develop, what it will look like in presence. So we are suggesting using restriction factors as a disease development markers for us to see where the disease will actually go, how long it will last, and how we can help. These restriction factors are cellular based proteins that basically inhibit HIV replication. We have 4 hallmarks of these restriction factors. One of them is virus restriction. Second of them, because HIV, he develops defense mechanisms, so these proteins fight against HIV's defense. Another one is interferon responsiveness, and the other one is inducibility. This all lead to restriction of the virus. This would be an ideal thing. What we've done, we have had 121 samples from 31 patients divided into 3 groups: slow progressors, rapid progressors, and general population of HIV positive. They were formed out of 19 male and 12 female, 14 Afro-Americans, 16 Hispanic. This is a cohort from New York Bronx. These are the genes we have measured. What we did, design of our study was we had HIV-infected cells, which we divided into 2 groups. In the first group, we have stained with different antibodies for phenotype, life and death tethering, and also activation. Then we ran our samples on flow cytometry. With the other group, we did isolation of CD4 T cells, and then we measured real expression of these genes on qPCR. Interestingly, we also took a look if we had any people who would be carriers of CCR5 delta 32 deletion, which is associated with African prostitutes that are resistant to HIV. Fortunately, we had 2 heterozygotes. Unfortunately, it wasn't enough for any clinical study. So our results were that here you can see positive or negative correlation. If in our long-term progressors, we have very low values of viral load, the cells go up there in pretty good numbers. The normal levels are around 1,000, 1,300 in healthy individuals. CD8 cells, they play a role just as compensation mechanism to the death of CD4 cells. Here we can see the levels of activation, which positively correlates with viral loads. And here we have also positive correlation of BST2 tethering correlated with CD4 counts. Tethering is a molecule that is actually protecting the cell membrane from entering the virus. We took a look at correlation coefficients between our restriction markers, and all of them correlate with one another. Although there is one exception. That exception is SAMHD1, because if all restriction factors correlated with one another, SAMHD1 would go the different way with CD4 activation and also CD4 count, which basically means the more healthy in terms of HIV infection you are, the more SAMHD1 is produced. We also took a look at the gender difference and the difference between the races. And as far as we can tell, there is no significant difference between genders nor races, which is actually a good news because we're trying to propose here a marker for prediction. So we want to have it as less complicated as possible. And finally, I'm getting to the results we generated. So here you can see early expression of restriction factors in between our haplotypes, and also early activation. This is in acute phase, and these results are in chronic phase. This tells us that in rapid progressors, there is very high expression of some of our restriction factors, whereas the activation is also pretty high. On the other hand, in chronic phase, there will be very or mediocre levels of restriction factor expression in rapid progressors, but very high levels of SAMHD1. Schlafly 11 makes exception here because he's associated with elite controllers. And what this all means, I would like to explain here on this scheme. You can think of rapid progressors as of athletes. For example, sprinters, like Usain Bolt. They're very motivated, which stands for activation, and they have heavy muscles, which stand for high expression of restriction factors. So these people are ready for a short, fast run. But HIV, it's a marathon. So it means that they end up running pretty fast. And this is where it all breaks down. And the more exhausted they become in a shorter run, the sooner they basically pass away. On the other hand, slow progressors are more like marathon runners. So they are distributing their energy and stamina and strength in time, which allows them to last a little longer. Actually, it's
please, if you can come to conclusions, maybe,
Yes.
because, thank you.
So conclusions. SAMHD1 deviates from the rest of the restriction factor group and is negatively associated with the markers of activation and fully non-independent on gender and race, which makes it ideal candidate for a new marker establishment of the disease development. And also, we've postulated a new hypothesis that claims that high expression of restriction markers is not always useful, and it depends on the phase, if it's acute or chronic. And we have made a protocol that will associate the levels of SAMHD1 with the disease progression, and also our findings will help physicians for the best individual therapy and better prognosis and longer life expectancy. Thank you for your attention.
Thank you very much for your presentation. Please come here. And are there any questions?
Excuse me, but there is a lot of results that you showed us. You are studying where? In Hradec Králové?
In Hradec Králové, yes.
You are studying?
Yes.
Yeah. And you did that study in Hradec Králové or in
No,
US?
I've done it in Washington,
You have,
DC.
how long you have been there?
I was there for 6 months.
6 months. And this is the result of the 6-month study?
Yes, because I started preparing this study when I was
During
here.
the study here?
Yes, and when I came there, I was just
No, I would
executing.
like to understand, because there is a lot of results. It's immensely packed with the results.
Yes.
So, okay, altogether the study is interesting, but that I would like to understand how you are involved in that.
Actually, if I can say, I was a part of the team, but I was making the design of the study, and I was executing all the physical work, but I had people who were assisting.
Okay, thank you.
One short question. Could you tell us a bit more about this SAMHD1 protein, what it does, what its function in T lymphocytes?
Yes. Actually, this protein was found out in monocytes, dendritic cells, and not very much in CD4 lymphocytes, which is very interesting, because what it does in these other kinds of cells, it's blocking DNTP, which actually disables the assessment of copied DNA of the virus. But it doesn't do it in CD4 cells, and that's why we can use it as a marker of predictor, because it's not actually restricting virus in CD4 cells, but it's correlating with other stuff that we can tell based on the production and expression of this marker where the disease will go. And based on this, we can also design the therapy.
So it's predictive in these cells only, yeah? That's
It
not
is,
functional.
but also other part is the inducibility of other restriction factors that we would like to use as the actual therapy.
Thank you.
Thank you.
So if there are no other questions, I'd like to thank you very much for the questions and thank you very much for your presentation.
Good afternoon. My name is Agata Jarmusch and I'm from Medical University of Łódź. I'd like to present the results of my project entitled The Anti-Transit Effect of the Mixed Mu and Delta Opioid Receptor Agonist Bifalin in mouse models mimicking diarrhea predominant irritable bowel syndrome. Diarrhea predominant irritable bowel syndrome is a chronic functional disorder of gastrointestinal tract, which significantly reduces patients' quality of life. The main symptoms of IBSD are abdominal pain, diarrhea, abdominal discomfort, bloating, and others. Due to complex and multifactorial pathogenesis, it's difficult to design an efficient IBSD therapy. And at present, this therapy is limited to alleviation of main symptoms. Bifalin is an octapeptide enkephalin analog with mixed mu and delta opioid receptors activity. And the majority of the studies concentrate on antinociceptive activity of this peptide. It has been shown that upon central administration, Bifalin produced 3-fold more potent analgesic effect than morphine and 7-fold more than etorphine. Due to mixed mu and delta opioid receptor agonist activity, Bifalin displays minimal addictive properties and lowered tolerance development when compared to other opioids. In this project, we evaluated the effect of Bifalin on gastrointestinal motility in physiology and IBSD symptoms mimicked in animal models of hypermotility and diarrhea. First, we assessed the effect of Bifalin on smooth muscle contractility in vitro, in the ileum and in the colon. Tissue fragments were challenged with Bifalin in concentration from 10 to minus 10 to 10 to minus 6 mol. Subsequently, we performed a series of in vivo experiments in which Bifalin was used at the dose of 5 milligrams per kilogram and injected intraperitoneally. This in vivo series of experiments included whole gastrointestinal transit test, colon bead expulsion test, fecal pellet output, and castor oil-induced diarrhea. All the procedures used in this project were approved by a local ethical committee for animal research. We found that Bifalin significantly inhibited smooth muscle contractility in vitro, in the ileum and in the colon, in concentration-dependent manner. This inhibitory effect was reversed by naloxone, a non-selective opioid receptor antagonist. In further studies, we evaluated the involvement of mu and delta opioid receptors in this inhibitory effect by using selective opioid receptor antagonist, beta-phenyltrexamine for mu opioid receptor, and naltrindle for delta opioid receptor. In vivo studies, we evaluated the effect of Bifalin on gastrointestinal motility after oral administration of colored non-absorbable marker. We observed that Bifalin prolonged gastrointestinal transit. Then we assessed the effect of Bifalin on colonic motility by measuring the time between insertion of glass bead into mice colon and its expulsion. We found that Bifalin inhibited colonic motility and remained active up to 45 minutes after administration. Here we show the effect of Bifalin in pathophysiology. We observed that Bifalin inhibited gastrointestinal hypermotility in mice model of stress, which caused this hypermotility. Then we assessed the antidiarrheal effect of bifalin after oral administration of castor oil. We observed that bifalin delayed the appearance of liquid feces after oral administration of castor oil when compared to control. This antidiarrheal effect was reversed by beta-phenyltretxamine, a selective mu-opioid receptor antagonist, and by naltrindol, a selective delta-opioid receptor antagonist. To conclude, we found that bifalin, as a mixed mu and delta opioid receptor agonist, through activation of those receptors, significantly inhibited gastrointestinal motility. And therefore, bifalin could become an interesting template for novel opioid-based drugs to be used in therapy of gastrointestinal diseases. Thank you for your attention.
Let me invite you here. And are there any questions?
Excuse me, I don't know, I'm not aware. Is there bifalin used in any kind of treatment in humans, or this?
No, it's not used in humans. It have been used in numerous preclinical studies in which it was measured its antinociceptive activity mainly. So in some research, it was some studies about the after ischemic use of bifalin, but not in the gastrointestinal tract, in the around the IBSD, around the gastrointestinal diseases.
Do you believe that the results of current studies or carried out studies in the preclinical stage give a chance to use bifalin in humans as well?
I don't think that bifalin could be used in treatment of disorders of gastrointestinal tract, but it has potential because it's a strong analgesic agent and it significantly reduces gastrointestinal motility. And what's important, due to mixed mu and delta opioid receptor activity, bifalin displays minimal addictive properties, and due to cooperation between those receptors, we observe some desirable features, like during.
Thank you. That's okay. Thank you.
Okay, thank you. Yes.
I have one question. Are these effects you observed on GI motility related to the central or peripheral mechanism?
Bifalin acts in the central nervous system. We know that, but we are not sure yet. But we will do it in further studies. If this inhibitory effect on gastrointestinal tract is related only to central nervous system or also to the peripheral nervous system, but we think that it's both. So we will check it in further studies by blocking peripheral opioid receptors and comparing the results.
Like metronidazole or CTOP.
Yes, dimethyl.
Thank you.
Thank you.
Enjoyable work, and also the subject of this work is very tough because patients are suffering from huge discomfort during IBS, and of course it's difficult to treat and to find out modalities for the treatment. And my point is to your in vitro studies here about the conditions of this water bath organ you did. How long did you manage, like it was like intestine put outside of the body into the organ culture? I mean water bath, not culture, right? So how long it may survive, that's the first. Second, what did you measure actually? It was number of contractions, or it was, because it might be in this part of the intestine, it might be related to different peristaltic and also non-peristaltic movements.
Can I continue? I can describe you the procedure, the whole procedure, as you.
Of course, not in details, but how long?
We extract the tissue fragments and we put it in a special conditions and the temperature of 37 Celsius degrees, and in Krebs solutions with electrolytes and glucose, which enable to tissue remain alive up to 1.5 hours.
Did you measure contractions?
Yes, tissue were placed between two electrodes and were stimulated by some electrical stimulators, yes, some electrical signals, and tissue were responsive. Then we measured the amplitude of tissue were placed between two fragments of yes, amplitude of length of the ileum or in the or the colon, the length, yeah, by measuring the tension of the tissue.
Okay. Okay, thanks.
Thank you.
If there are no other questions, then thank you very much for questions and for your presentation. So the next presentation is the influence on valproic acid on glioblastoma multiforme U87MG cells after knockdown of AKT3 and PICKCA genes.
Good afternoon. My name is Marta Pacholewicz. I'm a student in Medical University of Silesia. I would like to present my work about influence of valproic acid on glioblastoma multiforme cells after knockdown of PI3KCA and AKT3 genes. Glioblastoma multiforme is one of the most malignant tumors of human brain. Patients which are newly diagnosed have short survival time. We can't resect a whole tumor due to invasive nature with healthy tissue margins. This leads us to focus on new treatment methods and look for epigenetic mechanisms of gene controlling. Epigenetic changes are very important in gene controlling. Chromatins unpacking is very important in certain transcription of certain genes. One of epigenetic factor is valproic acid. It's used as an anticonvulsant drug. Inhibition of histone deacetylases is which belongs to valproic acid. Influence on intrinsic apoptosis pathway, extrinsic apoptosis pathway, and also inhibits angiogenesis and cell cycle and causes oxidative stress in cancer cells. PI3K and AKT signaling pathway plays a relevant role in in cells function, and its main particle, it plays a crucial role in transfecting signals from the outside of the cell to the inner part. Its main effector is AKT, which, when activated, regulates metabolism, metastasis, proliferation, apoptosis, and differentiation. We chose U87MG cell line, which is one of glioblastoma cell lines. We use it to compare our results from previous studies with a T98G cell line. We choose it because it's tumorigenic in laboratory animals. The aim of our study was to compare the influence of valproic acid after knockdown of AKT3 and PI3K-CA genes to viability, cell cycle, and inhibition of invasiveness of U87MG cells. Cells were cultured in modified EMAM in 37 degrees Celsius in 5% carbon dioxide atmosphere. Then cells were transfected with scRNA specific for AKT3 and PI3K-CA genes. If transfection efficiency was checked. Then after incubation, we treated cells with valproic acid of 0.5 millimole per 4 days. We received different groups of cells, and we performed cell cycle analysis, invasion test, viability test, and quantitative PCR to assemble genes associated with apoptosis induction. We received promising results. Cell viability was decreased in groups with knocked down genes treated with VPA, and in group treated just with VPA. Invasion index was also decreased in all tested groups. We compared two cell lines, a T98G cell line and U87 cell line. In T98 cell line, a population of potentially apoptotic cells was increased in all groups where we treated with a VPA. But in a U87 cell line, we haven't get any difference between control cells and we compared also the results from proliferative index. Here we have similar results in both cell lines, except the group with knocked down AKT3 gene, where was no changes in U87 cell line. One of the ways of measuring apoptosis induction is to measure mRNA of expression genes associated with apoptosis induction. In Caspase-3, expression of Caspase-3 was decreased in group with treated with VPA and with knocked down AKT3 gene with VPA, but in group with silenced PI3K-CA gene was no different comparing to the control cells. Similar results we we got from Caspase-8 expression. In Caspase-9 gene expression, we received very low gene expression in groups treated with VPA and with silence AKT3 and VPA group. In expression of BEAT gene, we received similar results where the expression was lower comparing to the control group in a group with a silence AKT3 gene and in group where cells were treated just with VPA. Summarizing, a combination of VPA and knockdown of genes AKT3 and PI3KCA decreases cells viability and invasion index in U87 cell line. In both cell lines, gene silencing and VPA treatment changes amount of potentially apoptotic cells and proliferative index. But we presume that lower gene expression involved in apoptosis induction may indicate the role of other cell death mechanisms causing lower size viability. Thank you.
Are there any questions from jury?
You carried out the experiment for 4 days, and you checked the expression of or the activity of the AKT and PIK3 after that time, or how did you follow that this transfection functions or not?
Whole procedure took us longer than 4 days. First, we seeded cells in plated wells. Then we incubated them for 48 hours, for 2 days, with sRNA specific for these genes. Then, after 2 days, we gave them
but you made sure that it was successful, and
after 4 days we extract RNA using a TRISOL procedure, and then we performed a quantitative PCR.
Okay, I have one question. Don't consider me malicious, but it seems there is missing one control group in your study, which will be transfection with the plasmid, whatever was your form of small interfering RNA, but non-specific to any of these two knockdown transcripts, to avoid any confusion about transfection as a modifying moment in your cell cultures.
Yes, we didn't perform such group. We just realized our studies on before papers. Thank you.
So if there are no more questions, thank you very much for questions and for your presentation. So the next work is titled The Localization and Function of ARC in the Neuronal Cell Nucleus by Adrian Schum.
All right. To nie jest na prezentacja. Good evening, ladies and gentlemen. My name is Adrian Schum. I'm from the University of Bergen from Norway, and I thought of presenting my work. I'm a medical student, and there I'm following my normal medical studies. I'm on my end of my 5th year, and I'm also following the medical student research program. And I'm going to present you my project, the localization and function of ARC in the neural cell nucleus. And this is a project I've been doing in Professor Clive Bramham's group. So let me start by saying that all the pictures that you'll see here are all my work. One of them is, for example, this animation. We'll come back to that later. Let me start by saying that our group is interested in this protein called ARC, also known as ARG 3.1, in short, activity-regulated cytoskeleton-associated protein. It's an immediate early gene, and it's involved in learning and memory-related molecular processes. Its main function is being seen in synaptic plasticity, and you can see that it's located in the nucleosoma in dendrites. And in this staining that I did with the C7 ARC antibody, you can clearly see that it stays all over the cell in small punctangles, also out in the synapses, if you would zoom in closer. A dysfunction of ARC has been seen in different diseases, and it could explain diseases such as Alzheimer's disease, Fragile X syndrome, Angelman syndrome, and other diseases. So the aim of this study was to identify nuclear binding partners to this protein ARC. So in collaboration with Professor Grzegorz Wilczyński and Kamil Parobczak at the Nensky Institute of Experimental Biology in Warsaw, they did a study there where they found some putative binding partners that they identified by pull-down and mass spectrometry. From those results, we found out 2 proteins that we were interested in looking further on, and this is where my part of the project started. So there we wanted to see the co-localization of PSF to ARC and SRRM to ARC. So PSF stands for polypyrimidine track binding protein associated splicing factor. It's been discussed that there could be several different functions of it, but mainly it's been seen in pre-mRNA splicing, RNA editing, DNA unwinding, in repair replication, etc. When it comes to SRRM2, it stands for serine arginine repetitive matrix 2, and it's been correlated to pre-mRNA splicing, and its cellular localization has been seen as being close to nuclear speckles. So this is something you can see in my staining, probably in this presentation. It's pretty light here. It's much easier to see on the screen here, actually. So when it comes to the study design, what we've done is that I've been working with in vitro cell cultures. So what we do is that we have a pregnant rat that we euthanize, and the embryos we take out and dissect out the hippocampus area. By using enzymes, we can, just to give you a general idea, by enzymes, we can then separate the cells and continue to grow them in vitro on slices, excuse me, on glass. And then we keep them alive for 13 days. And after 13 days, I stimulated the cells with BDNF, growth factor that has been seen previously to affect mainly the ARC, and in general, it's if you look simplistically at it, it's been seen often in synaptic plasticity. Further on, I did immunocytochemistry with ARC, PSF, SRM2, plus coupled with Alexa fluors. And the staining has also been done with prolonged gold that itself has DAPI. So that's when you mount actually cells. After that, I did confocal imaging using a confocal microscope and has optimal settings to get a nice set stack of 41 slices per each cell, called set stack, which represents the entire cell in a volume. So in general, you can see that these are just some examples of some of my cells. And if we look closer, this would mean that if you have one protein colored in green and another one in red, if they meet close to each other and emit yellow light, that could mean that there is a co-localization between these two proteins. But I did a different approach by doing this in 3D. And instead of using pixels, I used something called voxels. So you have X, Y, and Z planes. So besides the ARC staining and the protein staining, as in this case, PSF that was in red and ARC was in green, you also have the DAPI staining, which is staining all the DNA, meaning that it's actually showing you the nucleus. So I used the blue staining in order to mark out the nucleus, and in that way, I could show co-localization only in the nucleus. So this can also be seen in this presentation of the cell of what I could see when I was analyzing. So what you see here is that the blue color is then the nucleus. If you show you that again. The red is then PSF, green is all the ARC. And after removing and just taking out the nucleus, what we have left is the yellow, which would be the co-localization only in the nucleus. And all of these results has been done then in using voxels in 3D space. So in similar fashion, I did that for SRM2 and ARC. So you can see here where in normal 2D pictures, you can see where there could be a co-localization. So when it comes to the results, I saw that when it comes to the co-localization of PSF and ARC in the nucleus, in the controls, when we didn't simulate with BDNF, there was a very low co-localization, as it can be seen in the image here. But upon BDNF stimulation, there was a small increase in the co-localization of up to 20%. When it comes to the SRM2 plus ARC in the nucleus, when the cells were BDNF stimulated, there was a low co-localization. But the interesting thing was that the ARC puncta was mainly associated with the surface of SRM2 structure. So if you do a co-localization study, the results might be low, but around this SRM2 proteins, you could see small puncta of ARC. And to summarize that, this may be suggesting that there's a novel function of ARC related to its interaction with these paraspeckled proteins. But this is just a hypothesis that there might be something happening, but this hasn't been shown before. So to summarize my work, by researching ARC's function and its binding partners, we think that neuronal synaptic plasticity can be further understood. Do you have any questions? Yes.
First question, you told me that your Z stack of optical sections is 41 number.
Yes.
What is the total thickness? So what is in fact the thickness of optical section?
Section, well, it's, I know that I've optimized the I don't remember it correctly, but it's around maybe just 5 or 8 micromolar. So it's...
how thick is the cell?
The cell in this case are around 4 or 5 micromolar. Okay. Because it's between two slices. Of course, it's between two glasses. It's a bit squeezed down. But all these images, also the video, and this cell I'm showing here, it is between two slices, but you can still see it in a volume. And what's important that if you just take one slice out of that, you won't see the entire cell. Either you get some dendrites that are outside of the focal area, or you might miss some parts in the nucleus. That's why I think it's really important to get the entire cell in.
I found that you used double immunostaining. Did you use the primary antibodies from different species?
When it comes to ARC, we've been studying several different, we've been using different species for different experiments. In general, basically, we have a stack of around 4 or 5. There's not that many antibodies.
Yeah, but there are some techniques that allow us to use the primary antibodies from the same species, but the most common is just to use primary, for example, mouse and rabbit antibodies to label 2 different antigens.
That's what we did.
Like this.
I haven't explained it. I made it really a general presentation, but yes, of course, we use a different species for each antibody. So there's a lot of work put into that I put in order to find a good antibody that will work, and a lot of controls.
I was intrigued by your results on increased colocalization upon stimulation with BDNF. First of all, have you measured total of this ARC protein signal in nucleus before and after exposure to BDNF to be sure there is no nuclear localization induced by this trophic factor?
What we do see is that, so first of all, everything is in this case relative, and it has been put up in a relative setting, if I might explain, when it comes to the amount of protein. Yes, ARC does increase, and the transcription and translation of ARCs gets upregulated by
So
BDNF.
it's induced simply.
It is
by
induced,
BDNF.
yes. And that's something we know, and that it's common knowledge that it will happen. But it's necessary to start up the machinery to see the function of synaptic plasticity. What we're trying to do is to start up a chemical LTP, if you might know what that is, to start up potentiation of the synapses.
Okay, to do some functional studies of this, as I understand. My other question is, does the ARC protein, which is mostly located at the cytoskeleton, of course, and sometimes remote from the neuron body, does it have nuclear localization sequence for active transport across envelope of the nucleus, or is transported with some other protein?
And no, it has, as far as I know, it has yes, but it's also been seen, which I've seen in my stainings, that it's also the protein itself stains in the nucleus itself too. Yes?
Adrian, the question about this ARC, which is intriguing, of course, some functional significance of and quantification of of this is absolutely needed, because by immunocytochemistry looks very, very nice, but the question is about, if you're talking about the plasticity, then you have to, I think, give some numbers also. My point is related to, it was quantitative immunocytochemistry, but the functionality of this. My point is related to the expression of this ARC, which is actually at the baseline is low, whereas you have to stimulate, and you did with BDNF. The question is, the most common is nerve growth factor, right? NGF. So did you try to compare that, or I assume your prep was hypothalamic neurons, so you selected BNDF, but yeah, hippocampal. But have you tried with NGF? Perhaps is very good trophic factor, and also works not only at the central level but at the periphery as well.
Not NGF. What we've been using, what I've personally started using now is bicuculin plus 4AP, which has been shown in some papers, new papers that's come out, that it's a very strong it induces a very strong chemical LTP, long-term potentiation in these cells. So in some projects, we started using bicuculin 4AP instead of BDNF. But let me just say that why we started using BDNF is that my professor and this group has put a lot of work into BDNF, and also in order to compare to the results that previous people have had in the group, and to work together with the collaboration at the Ninsky Institute. That's why we chose to use BDNF.
Very short question. The last one. You showed us beautiful images, which I personally really like. What is this?
This is a normal hippocampal neuron, for me normal. Well, we can take this one, for example. This is also hippocampal neuron. What I've done here is, again, this is a cell that is grown on a glass slide. It's been fixed. So, and particularly this one is not alive. I've done experiments with using and following up a neuron
This is
live.
just one neuron from your control, from your
Yes, this is one neuron, and I've used a volume filling, in this case Dendra2, which is a fluorescent protein that is photo-switchable. The imaging itself, I've optimized so that I get the best resolution that's possible. So that what's actually blocking me is the optical resolution, what's physically possible to get a
I'm not
further
asking about the technical problems.
Yes, I understand.
Thank you.
So thank you very much for your presentation and from the questions. Thank you. So the next presenter would be Franciszek Sanak with the work CD56 bright natural killer cells respond to the in vitro stimulation with higher increase of cytotoxic markers expression than CD56 DAM NK cells.
Now it is working? All right. Ladies and gentlemen, it's a great honor to me to present you results of our research carried out on NK cells. NK cells represent a cellular part of natural immunity. Their characteristic phenotype molecules are CD56 and CD16 or no CD3. Activated NK cells express more 69 molecule. NK cells can effectively eliminate bacterial and viral infected and tumorously affected cells without prior sensitization. Based on CD56 expression density, NK cells can be divided into two main subsets, so-called bright and dim. The CD56 bright NK cells are characterized by higher proliferative potential, but very low cytolytic activity. They're supposed to be predecessors of CD56 dim subset, which has much higher cytolytic activity, but loses its proliferative potential. We worked with umbilical cord blood, which brings many benefits. Firstly, it is basically a leftover after labor, and it's rich in CD56 bright NK cells. In general, umbilical cord blood NK cells have higher proliferative potential, and they show lower risk of graft versus host disease. And last but not least, they show positive graft versus leukemia reaction, and it's something we'll be like. Unfortunately, immaturity of umbilical cord blood NK cells leads to their lower cytolytic activity, which has become a great challenge for us. As I said before, we have two main subsets. The dim subset can be represented by black belt karate men who have very low proliferative potential. And on the other side, the bright subset we can imagine as white belt karate kids who have the great ability to learn and to reproduce. We hypothesize that in vitro stimulation with interleukins 2, 12, 15, and their combinations should increase cytotoxicity markers expressions of NK cells. In other words, we would like to turn our white belt karate kids into an army of powerful Chuck Norrises. The whole procedure consists of several steps. Firstly, our colleagues from the Department of Obstetrics provide us with fresh umbilical cord blood, which we subsequently separate through lymphocyte separating medium and get an interface of mononuclear cells. Mononuclear cells are then sorted in magnetic field using negative selection. The magnetic particles pull all the cells aside, and all is left in the middle is a pure suspension of NK cells. I'm sorry. These NK cells we stimulate with interleukin 12 to 15 and their combinations. We also have a negative control. After 72 hours of cultivation, we stain these stimulated NK cells for the phenotype signs, for the activation marker, and for main immunoregulatory cytokines, interferon gamma and tumor necrosis factor alpha. Last but not least, we stain for main cytolytic agents, perforin and granzyme B. We study results and changes of expression intensity by measuring mean fluorescence intensity using flow cytometry. Because of the time limit, I'm going to present you only results after stimulation with combination of interleukin 12 plus 15. The graph shows you expression of CD69 molecule in both dim and bright subset before left columns and after stimulation. What you can see is significantly higher increase of CD69 expression in the bright subset after stimulation with combination of interleukin 12 and 15. We also observed significantly higher increase of production interferon gamma in both subsets up to 2 times, and significantly higher production of tumor necrosis factor alpha also in both subsets 2 times. And now please have a look at this. As we all know, the dim subset is the one with higher cytolytic activity, right? But after stimulation with interleukin 12 plus 15, the level of perforin production is similar in both bright and dim subset. All of this thanks to more than 4-fold higher increase of perforin production in the bright subset after stimulation. And most interestingly, the production of granzyme B is very low in the bright subset before the stimulation compared to the dim subset. But after stimulation, the production level is similar, not even higher, in the bright subset. All of this thanks to 18-fold higher increase of granzyme B production in the bright subset. Isn't it amazing? These data are completely new and unique, and they haven't been published yet. In conclusion, we proved significant activation and stimulation effect of the cytokine combination interleukin 12 plus 15. We also proved this combination to be the most effective for stimulating NK cells. And most importantly, we proved significantly higher increase of cytolytic markers expression in the bright subset after stimulation, which basically means that we managed to turn our white belt karate kids into powerful Chuck Norrises. These stimulated NK cells can find their clinical implication as a potential therapy of hematological malignancies because compared to bone marrow, they show low risk of graft versus host disease, and they show the positive graft versus leukemia reaction. I would like to thank my colleagues for support and you for the attention.
So are there any questions? Any questions from our jurors or from our audience? Okay, we have one question from Professor Sanak.
We are not relatives.
You think so? Actually, I wanted to ask you.
However, it's nice to see someone. It's quite rare surname in Poland. My question is, you used a cocktail, which was the most effective, 2 cytokines, but we know from immunology that it is possible to find something which stimulates production of both 12 and 15. Have you considered that it might be, I don't know actually, but might be IL-25, for example, or some upper registers of interleukins?
That's a really
so-called master interleukins of inflammation.
Right, this was a really good question. Actually, the combination of interleukin 12 plus 15 is based on our previous studies that were carried out a few years later. And we personally tested few combinations, and we found this one to be the most effective. And it is also really accessible, and we didn't see so much side effects as inflammations as well. And actually, we are testing the NK cells not even for the production levels, but also if they are functional. And so we let them grow in excelligence with tumor lines, and we see how actively they can kill tumor cells and so on. And this combination is again the most effective.
So if there are no more questions, thank you very much for your questions and for your presentation.
All right, thank you.
And also for the presenters, I just wanted to say that despite our best efforts, now
Yes.
it's either the timer visible or the full screen mode. So we can give you this sound after 7 minutes, and of course I'm monitoring it manually. But if you want to see the time passing, you can take, I don't know, a phone or some other kind. I'm really sorry for this technical problem, but we can solve it right now. So now we'll listen to the presentation of Mr. Zasulan. And it is titled Effect of MXS2 plus a polymer synthetic drug on behavioral reactions in old stressed rats.
Good afternoon, ladies and gentlemen. I'm from Kazakhstan. Don't close.
Okay.
With aging, the functional state of the immune and the nervous systems declines, and the regulation of the relationship between the systems gets impaired, resulting in loss on homeostasis and the increase in the risk of death. Therefore, we may conclude that impaired neuroimmunoendocrine regulatory mechanisms may play an essential role in the general body aging process, which reduces adaptive capacities and the stress resistance of the body with age. In view of the foresight, the purpose of this study is to expose the effect of new synthetic medicines on behavioral reactions in old rats. In the experiment, we used male and female albino outbred rats weighing 350 to 380 grams. For stage of the experiment, we conducted first set with old rats over 22 months, weighing 340 to 370 grams, and the second set with old rats of the same age exposed to immobilization stress, and the third set with old rats exposed to immobilization stress and treated with polyoxidonium, and fourth set with old rats exposed to immobilization stress and treatment with MHF2 plus polymer. There were 10 animals in each set. The animals with emotional stress were treated with MHF2 plus polymer at a dose of 50 milligram per kilogram of body weight. The drug with laboratory code MHF2 plus polymer was synthesized at the Bekturov Institute of Chemistry of the National Academy of Science of the Republic of Kazakhstan. The drugs were administered during 7 days, as the experiment were performed immediately after the completion of drug administration and in 1 week thereafter. The open methods, emotional stress was modeled by placing the rats into a movement restricting placebo box and for 30 minutes daily for 7 days. Behavioral reactions in vertebrates and in the open file, active avoidance reaction and passive avoidance reaction were studied in animals in all the sets of the experiment. The results effect of MHF2 polymer combination on the behavioral reaction in all rats exposed to immobilization stress. The results of the conditional reflex of active avoidance analysis provided in table 1 shows the MHF2 polymer positive effect on the training and the new skill learning process in all stress rats in the first experimental session. Yes, the number of correct avoidance reaction has significantly increased fivefold in the first step, fivefold in the second experimental session as compared to the previous session. Table 2, according to table 2, the time of staying at the light chamber compartment in the animals treated with MHF2 plus polymer has significantly increased compared to the results of the rats, but also to the results of the rats treated with polyoxidonium. Therefore, treatment with this drug inhibits the positive effect of weak intensive short-term stress on the development of the inherent reaction strong light avoidance in rat. Please, this is 274. This is 100. The result of what the maze test shows that the time for findings of safe platform in the rats treated with MHF2 polymer was similar to that in the rats treated with polyoxidonium. In the second experiment, in the first experiment, in the second experiment, all stress MHF2 polymer session, the time required to find a safe platform was notable better in the MHF2 polymer treated rats, what allowed concluding that the drug improves that spatial orientation memory in all stressed rat. As can be seen from the above, the MHF2 polymer regimen in the active avoidance reaction test has a greater effect on the memorizing process than the process of generation of memory trace in all stressed rats. The inherent whole exploratory behavior will not improve under the effect of MHF2 plus polymer, whereas the drug improves the spatial orientation memory parameters. I'll play this video for this question. No, next. Yes. This is a conditional reflex of active avoidance. He's filled errors and the light. These rats are using MHF2 polymer. Yes. Yes.
Thank you very much for your presentation. Are there any questions?
No questions.
If there are questions, there is a question from Professor Brzozowski.
It's interesting, but would you like to bring us closer to this compound? What is this polymer? What's that?
Polymer.
Yeah.
This is synthesized in the Bergdorf University, and professors and working with this. And the findings. This is polymer. MHF2. Yes. Yeah. This is
chemically is what?
As chemical as just okay. Yes. This is the MHF2 polymer. 1,2-ethoxetyl, 4-dimethoxyphosphorol, 4-hydroxypyridine. Yes.
Okay, so that means it has some potential in stress model, and the question of this sequence, like first you exposed animals to stress, and then medication was given.
Yes, yes.
Am I correct? Right?
Yes.
How about if you can treat animals with that first, and then expose them to the stress? Have you used such a combination, like first drug and then stress?
Yes, first drug and then stress. No,
Yeah.
then first stress, then drug. The second, the first experiment, and the second experiment in half drug. That's in half drug, and after his okay, just after has stressed.
So the normal order of this experiment includes this drug, this polymer given after stress.
Yes,
Right?
yes.
Right. And this memory, because you use in the abstract words like memory and orientation memory, can we just can you study this in detail, or it is just based on the symptoms of rat moving? Yes. And you count perhaps twitches or how the animals move or how many times?
Yes, yes, yes.
Some quantification, right?
3 methods. We have 3 methods: plexical box, water maze, and Zeppelin file. In this, the plexical box, 300 seconds, and the rats have his home, and his my English was bad.
No, you're doing fine. Okay.
Yes, yes.
Oh, okay. So it was.
This is after all stress rats, MHF2 polymer.
Right.
Is 274 seconds. It has. Is what?
Mm-hmm. All right. Well, fine. Thank you.
So if there are no more questions, then thank you for questions and thank you for your presentation. So we'll proceed to the next presentation entitled Changes in Expression of miRNA 103, miRNA 107, and caveolin 1 as the results of ileal transposition, IT, in Zucker, CRL, in Zucker rats. So it will be Mr. Długaszek.
Tak, działa. Good afternoon. My name is Michał Długaszek, and I have a pleasure to give you a lecture about changes in expression of microRNA 103, 107, and caveolin 1 as the results of ileal transposition in Zucker rats. This study was performed in Department of Physiology in Zabrze from Medical University of Silesia in Katowice. And our supervisors were Tomasz Sawczyn and Dominika Stygar. And the head of the department is Professor Krystyna Żwirska-Korczala. The first, what is the ileal transposition? It's a kind of a new metabolic surgery. Why metabolic? Because we can achieve the metabolic effect without reducing the surface of the gastrointestinal tract, as we can see in bariatric surgery. And the most important part of gastrointestinal tract in ileal transposition is a distal part of ileum, because there are many K and L enterochromocrine cells. And these cells can secrete, after the contact with a food, these cells can secrete many incretins, and it leads to enhanced insulin secretion, and finally, we can observe a decreased postprandial glucose level in the blood. So in references, it calls incretin effect. The caveolin-1 and microRNAs were in my topic of this presentation. So caveolin-1 is a small plasma membrane invaginations, which is involved in lipid transport, and it's a critical regulator of insulin receptor. And you have to know that caveolin-1 is a direct target gene of microRNA 103 and 7. And these microRNAs can bind to messenger RNA from caveolin-1. And finally, it leads to that protein assembly of caveolin-1 is blocked. So the aim of this study is to assess the long-term effect of ileal transposition on microRNAs and caveolin-1 gene expression in the liver. So our material were 20 Zucker diabetic rats. They are suffering from leptin receptor failure, and thanks for that, they develop many metabolic disorders, as you can see. In this study, we used 11 to 12-week male obese rats from Charles Rivers from France. In ileal transposition, we have to prepare distal part of ileum, about 50% of distal ileum, and then we transpose it after the ligament of Treitz. We also performed the sham operation. In this kind of operation, it's a control group. We performed only 3 anastomoses in these positions, in analogical places like in IT. So there are some photos from our surgery. General anesthesia induction in this box, and you can see some preparations. Okay, and it's the beginning of the operation. We have to, as I mentioned, we have to find the distal part of ileum, prepare it. Okay, and there is under this tissue is prepared ileum, and then we make anastomosis. The next step of this operation is to find the jejunum after the ligament of Treitz. We have to make some place for prepared ileum, and we have to make 2 anastomoses in those places. And it's the end of the operation. For the 6 months, we were collecting some blood samples, and we are performing oral glucose tolerance tests. And 6 months after the surgery, the left lobe liver biopsy was performed. Then we used the real-time PCR and ELISA tests for some assessments. And here's results. The first graphs show comparison of microRNA expression in rat liver after 6 months after the surgery, and microRNA 107 expression in IT group was significantly lower in comparison to sham-operated animals. But on the other hand, the expression of microRNA 103 was significantly higher in comparison to sham-operated. The next graph shows a comparison of the ratios of caveolin-1 to microRNAs. And the first ratio, the caveolin-1 to microRNA 103 ratio, was significantly lower in comparison to sham, but the ratio of caveolin 1 to microRNA 107 was significantly higher in IT group in comparison to sham. And I mentioned about some metabolic effect of this operation. And here's a proof. It's a comparison of triglycerides levels. And what is confusing, 6 months after the surgery, the triglycerides levels was significantly higher in IT group in comparison to sham operated. But the low density lipoprotein level was significantly lower in IT group, just like for total cholesterol level, as you can see. And the last graph is a comparison of oral glucose tolerance test 6 months after the surgery. And the red line is for IT group. Area under this curve was significantly lower in comparison to another. It means baseline, animals before the surgery, of course, sham-operated animals, and animals without the operation. And finally, let me make some conclusions. IT improved glucose tolerance, and IT improved low-density lipoprotein and total cholesterol metabolism, and IT affects the gene expression in a liver. But there were some conclusions in long-term observations. It was the first study which shows these results after 6 months. And today, I can suggest you IT as an alternative surgical method of treating type 2 diabetes mellitus. But you have to remember that life is like a box of chocolates. It doesn't last long for obese people. Thank you for your attention, and let's discuss.
So are there any questions? There is a question from Professor Sanak.
Technical question, because you
yes,
measured the expression of 2 microRNAs belonging to the same family, 103, 107. Why you observed this
differences, yes?
effects? Yeah. How would you explain? I saw also that they differ in their concentration by more than factor of 10, yeah, or even more, 100.
I can explain this in references. I found that the microRNA 107 red boxes is much more associated with caveolin 1 than microRNA 103. And in our study, we found that dose ratio can block the assembly for much higher effect. Yes.
So affinity of one molecule is higher than the other.
Yes.
Methodology was clearly shown. I enjoyed this. And my point is regarding the specificity of this data. If we compare this Parisian Zucker rats with others, perhaps Streptozoetis in rats can also be used to demonstrate a high level of diabetes, right? So my point is, you think it would be the same, or I think microRNA might be different, right? Under different experimental setups. That's the first question. And second is regarding this operation by itself. I think it's not easy then to keep animals like that. So you have 100% of survival after that, or
no.
Not really, right?
Not really. 2 rats were died in this study, and we didn't include it. And the first question, And now we are working with Sproge Dowry rats, and we will see how it results. It will be in this kind of rats. We have to check how the glucose tolerance and insulin resistance will be in the others models, because this kind of surgery is performing in the world.
The last question, yes, from Dr. Gaida.
One question: how many animals have you operated?
I operated about 70.
70. How
70,
many?
yes.
What was the percentage that survived the observation period?
At the final stage, I think about 90%. But in the beginning of my work in the Department of Physiology, it was about 60%.
Thank you.
I don't remember, but.
So thank you very much for questions, and thank you for your presentation. So for our last oral presentation, we invite Aleksandra Dudzik with the work Do the Statins Reduce Prostate Cancer Progression?
Good afternoon, ladies and gentlemen. My name is Aleksandra Dudzik, and I would like to present today my and my friend's Kamil Grabowski study entitled Do the Statins Reduce the Prostate Cancer Progression? Our work was conducted under the supervision of, and with a great help of our tutor, Dr. Joanna Dolińska-Litewka. So let's start. Prostate cancer is nowadays a very important clinical issue. According to the statistical data, it is the most common cancer in men in developed countries, also in Poland, and the third one if it comes for mortality. Well, prostate is affected by a lot of different steroid hormones, but especially the dihydrotestosterone and its receptor, the androgen receptor, play a very important role in the growth and differentiation of the cancer cells. Unfortunately, due to ineffectiveness of androgen deprivation therapy in androgen-independent prostate cancer, lately an urgent need arose for the new medication. And that is why attention was dragged to the prostate cells' cholesterol metabolism, and in this way also to the statins. But what are the statins? Statins are widely known drugs commonly used for the treatment of lipid disorders. As you can see on the presentation, their main action is inhibition of 3-hydroxy-3-methylglutarate coenzyme A reductase, a regulatory enzyme of cholesterol biosynthesis. In this way, statins are believed to reduce the level of circulating dihydrotestosterone, as it is synthesized from cholesterol and is also a main ligand of androgen receptor. Another way of statins' impact on the prostate cell metabolism are the so-called pleiotropic effects that are mediated via the inhibition of protein isoprenylation, a process that is crucial for the activity of pro- and anti-carcinogenic signaling pathways in the cell. But what was the aim of our study, actually? The aim of our study was to check if inhibition of androgen synthesis in the prostate cell can reduce the growth of the tumor and also reduce the risk of prostate cancer metastasis. Our study was conducted on the 4 prostate cell lines, 1 normal, PZHP-FAO, and 3 cancer lines, LNCAP, PC3, and D145. As inhibiting factors, we used two different statins, simvastatin and fluvastatin, and we investigated their impact on the expression of the following proteins: androgen receptor, AKT, caspase-3 and 9, also P21 and P27, matrix metalloproteinases, and beta-actin. Laboratory techniques we used were cell culture, cytotoxicity analysis, gelatin zymography, proliferation assay, also invasion assay in Boyden chambers, and Western blot analysis. Now, let us move to the results. First, cytotoxicity. We measured this actually undesirable effects of our inhibitors by incubating the cells with simvastatin and fluvastatin in different concentrations. Actually, no significant cytotoxic effect was observed in every concentration, both for simvastatin and fluvastatin, what is actually something that we anticipated. Then proliferation. We used the crystal violet test to assess the number of the living cells in our probes. We used different concentration of both fluvastatin and simvastatin in 4 different cell lines. As you can see on the diagram, you can see the percentage of the living cells in comparison to the control without the stimulation with the statins. The greatest effect we observed in cancerous LNCAP line and the least visible in the normal PZ-HPV line. The same tendency we observed for simvastatin, whereas the greatest effect was for the LNCAP line also, and it was an inhibition of proliferation from 11 to 78%, depending on the concentration and the time. Then, gelatin zymography. Here we analyzed the activity of matrix metalloproteinases, the enzymes that are responsible for the metastatic potential of the cells. Here you can see the photos from our gels from zymography, and you can see also the narrowing of the white stripes in the probes where the material from the cells stimulated with the cells was used. Actually, the narrowing of the stripes means that our inhibitors really worked. I mean, they inhibited the activity of metalloproteinases and also the risk of the metastasis in the same way. Then, the invasion potential. As you can see in the table, there was a strong dose-dependent tendency of decrement of invasion potential in these 3 cancerous lines, both for simvastatin and fluvastatin. Then, the most important part of our work, actually, the Western blot analysis. Here you can see different proteins on the left, and the stripes that are, their thickness is actually proportional to the expression of every protein. Here we observed actually what we anticipated. I mean, the androgen receptor and phosphorylized AKT, the proteins that have a pro-carcinogenic activity, where their expression was downregulated, whereas caspase-3 and 9, P27 and P21, with anti-carcinogenic effect, all 4 were upregulated. You can see the greater thickness of the stripes with the stimulation of the statins in comparison to the control. The same effect we can observe for fluvastatin. Now, moving to conclusions. According to our results, statins significantly reduced the proliferation of the prostate cancer cells, also diminished the metastatic potential, the invasion potential of the cells. They upregulated the signaling proteins that are responsible for the anti-carcinogenic effect in the cell, and downregulated the expression of the ones that work pro-carcinogenic. Such conclusions encourage us to the further investigation of the androgenic pathway in a cell, and also give us hope for, of course, the successful use of the statins in the future treatment of the prostate cancer. Thank you very much for your attention, and at the end, I would like to thank and express my gratitude for the whole Chair of Medical Biochemistry, especially for our tutor, Dr. Joanna Dolińska-Lisewska, for enabling us conducting this study and a great help and supervision. Thank you very much, and I'm waiting for your questions.
So I invite you here for questions. So are there any questions from our jurors?
This was absolutely one piece, very good. And question for this apoptosis, that sounds very strong. Like you have this caspase family, I understand, upregulated, right? Which means our apoptosis might be increased, and it goes along the line that it might be anti-carcinogenic. Both of them. My point is related, if you can strengthen that, looking for expression of BAX and BCL2 perhaps, are they changed? Have you ever tried this? These are pro-apoptotic and anti-apoptotic proteins.
Yes.
I just wonder, because you don't have to.
Yes, I know the protein BCL2 and BAX, you mean. I know that they also play a very important role in a prostate cancer, but we didn't investigate it so far. But maybe in the future, why not take it into account in the next study?
There is also atorvastatin, another form of, not tested yet, but it's, well, that's the fresh idea that maybe all of them, all statins are working the same way, right? Can we say so?
Yes, why not? We can try atorvastatin next time. This time we chose simvastatin as a natural statin and pravastatin as an artificial one as a comparison.
Right, thank you.
Thank you.
So if there are no more questions, then thank you very much for your questions and thank you very much for your presentation. And now I would like to invite you for 15 minutes break before the electronic poster session. So let's meet here about maybe 10 minutes, okay? If it would be great if you can be here again in 10 minutes. Thank you very much.
Great, thank you.
Evaluated together. Yeah, but who is the head party? Who is the head?
No bo tak jak mówiłam, w pewnym momencie mamy trzy.
To nie można mówić po angielsku, znaczy po polsku.
Idę na liberalization, wiesz, bo po prostu hemoroidy mnie bolą.
These are the poster sessions or they really? Okay, ładnie, okay. Tak? A co z tą prezentacją tutaj?
Nie, reszta będą postery elektroniczne.
Nie, ja rozumiem, tylko że to są tylko...
Tych nie ma.
I dwóch dalszych
Oddamy razem, oni wiedzą, co z tym zrobić. These two will not be zrobione. No, one before. One before. We keep this for
Okay.
So how are you, Maria? Are you in love? You slim down, looks well. Okay.
Myself, I guess.
Really, as
Thank
usual.
you. Thanks. How's everything going with?
A lot of teaching, young Norwegian students,
Yeah, eager
first
to
year,
learn.
freshmen.
Yeah.
Kids are rude, as they say.
They're
Just childish.
That's the stage that teachers are in. I remember when I was in my first year.
They need some, training.
Yeah,
Because they-
but does it depend on the group? Like,
on personality of the student. Some are more serious than others are not. Evidently, they had very bad session of psychology.
really? It's kind of like it follows the same pattern, like, depending on if they how they do on the other
Highly
exams.
replicable.
Yeah. And you've seen it all.
Nie, nie.
Bo jeszcze nie jestem po IT Operation.
Bardzo dobre.
U nas na przykład troszkę zwagi jest też. Oceniamy przedsiębiorstw.
Prawdopodobnie są przyczyną, że jest to dzieło w momencie.
Policzyliście już to? Jest. Nie ma rozumienia, że tylko telefonowy, że jest kontakt, bo żeby pracować na komputerze potrzebuje kontaktu.
And as well, this is your second.
Yes, I think I will move the account response. Yes. sorry, it's just switching. The PDF is here. I use this to introduce. But here you have every poster. I had to switch in between presentations because yes, because of the PowerPoints. But here I have everything. But let's try. Let's try again with this timer because I have no idea why it shows on some of them, and as you see, it's gone. I have no idea how it works. Why is that?
We had that. You have to tell us results now or after the poster session?
After the poster session, because the competition is together. It's like is together for posters and for time.
Also, there are going to be only 3 medalists in the whole 24.
Yes,
or
and two distinctions.
Yes.
So like 5 prizes.
We can fix it now.
From the whole 20.
Yes,
We
yes.
can
5
sit.
prizes.
Well, it's time for presentation.
2 minutes.
2? No, 4 maybe. 2 minutes for discussion.
Marking it, and most of them are connected with the work and not the way of presentation. And there is one category which is connected with the style and way of presentation. And so here, and in each category, jurors have from 0 to 10 points. So there is a category for presentation, and there they can take into account, you know, that it was poster or it was said badly or something.
And when does it start? The presentation, I mean, the posters.
In 5 minutes. In about 5 minutes, yes.
And then the results about 7 p.m.
Yes, yes, yes. After the end of all the posters, we will sum up everything. Actually, I will start summing up now, and then we will get results from posters. We will probably need like 15 or at maximum 20 minutes to have the final results.
Right.
Yes, and also, you know, we are thinking that the same categories and much more time for oral presentations. It's like, of course, papers which were better in review, they got oral presentations and slightly worse as electronic posters. So if you have nearly twice as much time for presenting your work, you will be probably much more convincing, yes, to the jury, and have a chance to really tell more about your work. So, but the competition is like for all French people.
Right. Yes, thank you.
Chciałabym przywitać do 17. Także
Dobra, to
podałyśmy
sorry za
kawę.
przepraszanie i bardzo
Nie wiem, czy
dziękuję.
dobrze położyłyśmy po prostu, bo tam dla kontraktora. Tak, Sara. No, to raczej chyba się powinno miejsce zgadzać. Jeszcze tam jeden pan chciał wodę, to wziąłem z satu.
Super, dziękuję bardzo.
Właśnie.
Na którym głosujecie? Po elektrycznym.
Po elektrycznym. Koło 7 będzie.
Około 7 będą?
Koło 7.
Aha, no to dobrze. A, to
No
sobie
bo,
przejdę na pół smykacze.
Koło 7. No bo myślę, że zaczniemy tak za 15, zanim wrócą wszyscy profesorowie tak realnie, to myślę, że za 15.
Okay.
No i te postery powinny się zamknąć grubsza w godzinę, 10 minut, czyli no koło 7 może się zdarzyć, że skończymy sesję i my to podsumujemy.
To sam raz
I tyle.
na autobus także.
Mhm.
Dziękuję.
4 minutes? 4 minutes, yes. And I was... No, but I don't think... I was just trying also to, you know, manually write down, you know, yes, just the time, so you can read. And I will try again, but actually this one is very strange because, you know, it doesn't react for anything here. I don't know why, but it is not reacted. It didn't before. Control L, yes. So Control L. And like this. And how do you open that? So this one should give you a sound signal. It's like here on timeout, play sound. So it is set now, yes. And then I will try to start it and now. Okay, and as you can see, it is gone now, but it will give this sound signal. So, and you have to look at the watch. I have no idea why it will say that. And also, let's try.
Z czasem tak ostro, bo rektor musi na galę zdążyć.
Ja wiem. Nie, do 19 na pewno jury będzie wolne, a później to my
Tak,
sobie
będzie?
punkty, a oni mogą iść. Jest 9
Nie,
plakatów.
oni też chcą, wiesz, ogłosić.
9 plakatów.
Więc trzeba teraz podliczyć, no?
Tak,
Biorące
ja będę
trzeźwość.
podliczać, to ja będę z tym
No,
tylko...
żeby oni tak 19:10 max skończyli
Każdy
wszystko.
plakat to jest 6 minut, wiadomo, to jest jakieś przejścia ludzi, no ale...
Że
myślę,
czasu
że-
ucinajcie, jak
Dobra.
ktoś będzie.
Dobra.
Po prostu ucinajcie.
And as you can see, with Control Plus and Control Minus, they can zoom in, zoom out. Yes. So actually, you can announce it, at the beginning of the presentation, so everyone will know that they can sit here or stay here, and with this they can zoom in, zoom out. Yes, because I don't have any better method if someone wants to zoom anything. And here we have the list, so if there's anything else you need, just click on it. And just not so will they have to zoom in or should I zoom in? No. It should be like that, and if they want to, they can say the presentation from here and manually just if they want to. Yeah, if they need to, but maybe this size is okay, yes? And okay, so this works too. So you can have to leave the house. Here, it works okay.
All right, so we've come to the second part of our session tonight, and it's going to be on the electronic posters. Now, the timing for the presenters is 4 minutes, and you'll be able to have commentaries for 2 minutes after that. And if you need to zoom into your posters, you can just do that manually. Come over here and zoom in if you do need to do so. And also, there's a time to indicate when your time is up. So. The first presentation is by Strobel Spencer, and it's on antidepressant drugs modulate humoral immunity in mice via the inhibition of antigen presentation by macrophages.
Where's the... And is there a laser? Although there have been several studies in the role of antidepressants and decreasing inflammation in patients with depression, there's very little known about how antidepressants can affect our humoral immune response. Recently, in previous studies, we established that fluoxetine, venlafaxine, and meflodomide reduce cellular allergy, whereas
Okay, awesome. Okay, whereas imipramine slightly enhanced the secretory slightly enhance the immune response. So the aim of our study was to examine antidepressant effect upon humoral immune response, and in the same we used macrophages from donor mice that we treated for 7 days with antidepressants, and after we treated them, we tested them, the ability for them to invoke the humoral immune response. And we did 3 different studies. We tested in a plaque-forming assay the levels of splenic antibody-producing cells, as well as we looked at the antibody titers in the sera, and we cytometrically examined the cell surface markers of the macrophages recipients. And as you can see here, we observed in fluoxetine, venlafaxine, and meclobamide, we saw a decrease in the plaque-forming cells, or the splenic antibody-forming cells, as well we saw a drop in the antibody titers in the sera in each of these 3 antidepressants. Conversely to this, in imipramine, we saw a slight enhancement of, as you can see here, and then enhancement of the humoral immune response. Interestingly, in the venlafaxine and fluoxetine treated mice, there was an increase in the maturation of the humoral immune response. As you can see, there's no IgM antibodies. They're all IgG. In the cytometric study, in the flow cytometry, you can see that in examining the different cell surface markers of antigen presentation, and as well as phagocytosis, we noticed that there's a reduction within, again, fluoxetine, venlafaxine, and meclobamide within these cell markers. And in conclusion, our studies demonstrated that there was a reduction in the humoral immune response, and in the fluoxetine, venlafaxine, and meclobamide treated mice. And there was, in contrast to that, an increase in the imipramine treated mice. And we can say that this was due to the modulation of the antigen surface markers, or the surface markers for antigen presentation and phagocytosis. And so this may lead to an impaired antimicrobial immunity, but we theorize that with this treatment with treatment of antidepressants, we can reduce the level of depression and level of other symptoms within humoral allergies, as well as in people with autoimmune diseases. Thank you for your attention.
Thank you, Strobel. If you'd like to come behind and use this microphone. And if the jury have any questions.
Are there any questions from jury or from the audience? Okay. If there aren't any, I just wanted to remind you what my colleague has said before, that if you want or need to zoom any part of your poster, you can just use one of your stands with the computer to tell about your work, and then use just Ctrl+ or Ctrl- to zoom it.
All right, so we'll go on to our next poster. It's by Polina Kowalczyk, and the topic is on diet-induced obesity, aggravates natural killer cells mediated contact hypersensitivity in RAG1 B6 mice.
Contact hypersensitivity is a form of delayed type hypersensitivity and is mediated by antigen-specific CD4 or CD8 lymphocytes T. Previously, it was shown that NK cells can play a role of effector cells in CHS. Also, recent studies have shown that diet-induced obesity aggravates classical T-cell mediated contact hypersensitivity. So the aim of my study was to determine the influence of diet-induced obesity on NK cell mediated CHS. NK cell mediated CHS was induced in mice genetically modified lacking T and B lymphocytes. To induce obesity, mice were fed with a high-fat diet for 2 months prior to CHS induction. Control mice received normal diet. Animals' body weight were measured once a week. After 2 months, mice were sensitized with 0.5% DNFB, and after 5 days, CHS responses are elicited by painting both ears with DNFB. Ear thickness was measured before challenge and then 24 hours after challenge. Ear weight and myeloperoxidase activity in ear tissue were tested 24 hours after challenge. Unsensitized mice, both on a high-fat diet and normal diet, were used as a control. To perform biochemical analysis 24 hours after challenge, the blood was collected and then the serum was separated from the blood. Now let's look at the results. How you can see, mice fed with a high-fat diet gained significantly more body weight compared with mice received a normal diet. Biochemical analysis showed that high-fat diet increases total cholesterol and low-density lipoprotein concentration in RAG1 null mice. When you look at the third graph, you can see that obese mice showed significantly increased ear swelling response compared with lean mice. Increase in ear thickness correlate in mice received a high-fat diet, correlate with higher ear weight and increased MPO activity. So the main conclusion of my study is that high-fat diet aggravated natural killer cell-mediated contact hypersensitivity in RAG1 null mice. Thank you for your attention.
Thank you, Polina. Now, if the jury has any questions, we'd invite you.
That's very interesting. Have you evaluated the total number of peripheral NK cells in these animals who gained weight?
No, not yet. But in further experiments, I want
because
to.
A possible explanation could be,
only,
some proliferative signal from
yes,
fat.
but for example, I examine the fat tissue, and I see that interleukin 6 is increased and interleukin 10 is decreased in fat tissue, visceral fat tissue.
Thank you.
So I'd like to invite Dean Jabrovic. And the topic that Dean will be presenting is assaying biochemical parameters of liver function in vivo induced oxidative stress and after application of Thymus panonicus, it's a bit of a mouthful, extract. Right, so.
So, honored members of Commission and dear audience, it is my immense pleasure to present you. Where is it? Oh, it's here. Thank you. So it is my immense pleasure to present you results of our work by the name of assaying biochemical parameters of liver function in vivo induced oxidative stress and after application of Thymus panonicus extract. So chronic liver diseases are an enormous problem all around the world. Standard medicinal therapies are often limited in their effectiveness and also can carry a risk of side effects, and also can be very expensive for countries in development. So usage of medicinal herbs is one of the alternative solutions. Eurasian thyme, or Thymus panonicus, is perennial herbaceous plant mostly distributed in Central Europe. It is known in traditional medicine as a therapy for coughing and other respiratory problems. But in this scientific work, we were examining whether or not this plant, precisely its extract, has any hepatoprotective effect. So, at first, we were doing hydrodistillation, which is the most suitable way of producing essential oils. And during this process, always remains as a waste product this mixture of herb and water. And it was really interesting for us because this is always considered as a waste product. So we made extract of this waste product to see if this has any hepatoprotective activity. And we were doing that in vivo systems. Our in vivo systems were Sprague-Dawley rats divided in 4 different groups. First group was the control group. They were given during the 7 days 0.9% sodium chloride solution. Second group is the negative control group. They were given the same treatment as the first, but 24 hours before sacrificing, they were given carbon tetrachloride to induce oxidative stress. The third group was extract group. They were given different doses of Thymus panonicus extract. And the fourth, the last group, they were given the same treatment as the third group, but 24 hours before sacrificing, they were also given carbon tetrachloride, the same to induce oxidative stress. After sacrificing all animals, suitable biochemical tests were performed, and we were measuring body mass, liver mass, total content of protein, bilirubin, hydroxyproline, activity of AST, ALT, and hepatic DNA. So oxidative stress induced by carbon tetrachloride notably damaged liver functions and disrupted all biochemical parameters. But it is important that in groups which were primarily treated with Thymus panonicus, it is noticed significant conservation of body mass, liver mass, total content of proteins, and hepatic DNA. Also, it is noticed significant decrease of hydroxyproline, bilirubin, and activity of AST and ALT. So we can see that Thymus panonicus extract has annulled all these effects that were caused by oxidative stress. And in conclusion, we can say that Thymus panonicus extract could be used as a potential additional therapy in numerous liver diseases. Thank you.
Thank you, Tijan. Now, if any of the audience or if the jury would like to ask any questions.
What was vehicle for this extract? What was water solution or ethanol solution?
That's a water solution because after that hydrodistillation is hydrodistillation, so remains water and the plant. So we made extract of this equation. This is aqueous extract.
Okay,
actually.
thank you.
Good talk. Talking about this oxidative stress, it would be nice to have some, at least free radical measurement in the liver. Have you ever tried this? It is possible to manage this, like to see which particular free radical is contributory to this damage by CCCL4.
Well, we were measuring this oxidative stress only through this biochemical
All
parameter,
right,
so unfortunately I can't answer it.
All right. So among the parameters is also the sodium dismutase, which is an enzyme which can scavenge free radicals and oxygen, reactive oxygen species. So it might be additional parameters measured also.
Yes, I understand, but this is like a great project on our faculty and everyone is examining something else. So this was like a small part of examining this plant on biochemical chair.
All right, thank you.
Okay, so thank you. And I'd like to invite our next speaker. Oh, sorry, before you go, please present. Our next presenter, if you'd like to come up. Now, the topic is the prevalence of AAP, BHP, and ATLE genes among biofilm-forming and non-forming clinical strains of Staph epidermidis.
Thank you. So, dear professors, dear fellow students, as a representative of the Student Scientific Circle at the Institute and Department of Microbiology and Virology at Medical University of Silesia, I would like to present a part of our research concerning the prevalence of genes AHP, BHP, and ATLE among clinical strains of Staphylococcus epidermidis, forming and not forming biofilm. Staphylococcus epidermidis is one of the most frequently isolated opportunistic microorganisms. Despite the fluviolence factor, a high level of pathogenicity results, among others, from the ability to form a biofilm. Biofilm is a cluster of closely connecting bacterial seals immobilized by extracellular matrix produced by themselves. The aim of our study was to determine the prevalence of AHP, BHP, and ATLE genes responsible for biofilm production among clinical strains of Staphylococcus epidermidis. The ATLE protein express autolysin activity and is responsible for the initial phase of staphylococcal adhesion to the biotic and abiotic surfaces. The AAP protein is associated with the second stage of biofilm formation. It means accumulation. And the BHP protein is present on cell surface and initiate an alternative mechanism of biofilm formation. The study comprised 92 clinical strains from the collection of microorganisms of Instituted Department of Microbiology and Virology. To confirm the presence of genes, PCR was performed using specific primer pairs. Our result of phenotypic studies showed that 63 out of 92 strains demonstrate the ability to produce biofilm. Among the studied strains, the presence of AAP gene has been shown in 37 strains forming a biofilm, and in 15 strains do not forming a biofilm. The presence of BAP gene was confirmed in 22 strains forming a biofilm, and in 4 strains do not forming a biofilm. And the ATLE gene, the presence of ATLE gene has been shown in 63 strains of biofilm, of strains producing the biofilm, and in 23 strains do not producing the biofilm. Taking into account strains having ATLE gene, 51 of them also show the presence of AAP gene, and 24 the presence of BAP gene. In conclusion, I would like to stress that the most of biofilm forming strains reveal the presence of ATLE gene, which may suggest its significant role in biofilm formation. In the future, we plan to cast more light for the mechanism of biofilm formation. Thank you for attention.
Thank you. And now if the jury has any questions, please feel free to, yep.
My question is about practical value of your findings. Does it mean that, for example, someone scheduled for orthopedic replacement therapy should be taken swabs and analyzed for presence of colonization with this type of ATLE positive streptococcus?
Yes, but I don't understand good the question, if you can repeat.
My question was, can we foresee practical value of this finding in
Yes,
prediction of failures in implant healing or whatever?
I understand now. Staphylococcal biofilm is a structure which is produced at, as you said, orthopedics replacements or swabs, and continue our research will in the future help to eliminate the problem of biofilm at replacements or swab, for example.
But can we inhibit this autolysin, or we shall eradicate the bacteria rather?
We don't analyze this. Now we checking is ATLE responsible for sure for biofilm formation, and then in the future we will check how to inhibit this protein.
Okay, thank you.
Any questions?
Thank you.
Our next poster presentation will be by Mathieu, and the topic is quite simple, so content of chi-urenic acid in pasta.
Thank you. So the subject of my poster is quinurenic acid. Quinurenic acid is a metabolite of tryptophan obtained in quinurenic pathway. It was firstly discovered in 1853 in the urine of the dog. Quinurenic acid is interesting. Quinurenic acid is an interesting substance because it is the one only known antagonist of amino acid receptors in a central nervous system. It is antagonist of NMDA receptors, AMPA receptors, and kinic receptors in brain. It is also antagonist of alpha-7 nicotine cholinergic receptors that are presented in lymphoid tissue and in brain, and it is also agonist of GPR35 receptors that are in enterocytes and in mucosa-associated lymphoid tissue. The first researchers about quinurenic acid were conducted in brain. We noticed that, for example, we can determine more quinurenic acid in brains of people suffering from schizophrenia or bipolar affective disease, whereas lower contents of quinurenic acid are present in brains of people suffering from Alzheimer's disease or Parkinson's disease. Quinurenic acid has also function in peripheral system. For example, in digestive tract, we know that quinurenic acid have anti-ulcerative function and anti-inflammatory function. Also, quinurenic acid is antagonist of homocysteine, that is, according to newest researches, a factor that may lead to hypertension, to diseases of metabolic syndrome. Quinurenic acid in central nervous system may be obtained only from endogenous sources, whereas in digestive tract we may possess quinurenic acid from bile, from pancreatic juice, and also from food. It were conducted researches about content of quinurenic acid in food. So we know that higher substance that food rich in quinurenic acid are, for example, vegetables, fruits, honey, and also herbs, especially elderberry flower, for example, birch leaf or nettle leaf, whereas poor in quinurenic acid are sunflower oil, chips, or red meat. Bearing in mind this fact and the fact that Mediterranean diet is commonly known as a healthy diet because people who live in regions of Mediterranean Sea suffer less frequently from hypertension, from neoplasms, from diseases of metabolic syndrome, I decided to determine the content of quinurenic acid in pasta, the main ingredient of Italian Mediterranean cuisine. So here I had results of this research, and it turned out that healthy properties of pasta doesn't depend on the content of quinurenic acid in it, because the content of quinurenic acid in Polish pasta and in Italian pasta is quite small. But what's interesting, I noticed that, for example, we have a very high content of quinurenic acid in pasta Graham Świderki that was made from wholemeal flour. So the conclusion of this research was that the content of quinurenic pasta in the content of quinurenic acid in pasta depends on the type of flour used in producing the pasta, and maybe the pasta made of wholemeal flour is the most healthy. Thank you.
Thank you. And if the jury has any questions, again, please feel free to ask.
Flour. Pardon.
The preparation procedure for this material before analysis was not presented exactly.
Yes,
it's just
I'm gonna
written
tell it
in abstract. It was according to manufacturer's guidelines. But my question is, the substance is well soluble in water. During boiling, I assume you boiled the pasta before analysis. Have you checked how much of quinurenic acid is dissolved during boiling?
during boiling.
During preparation, because you're
unfortunately, we haven't measured it, but we know that the temperature that quinolinic acid melts is something like 280 degrees.
But
so we
it's
boiled
soluble.
this pasta in lower temperatures.
Just one single question, because if you look into metabolic path of tryptophan, right? So before the end leading to the quinolinic acid, you do have also sometimes melatonin, right? So is it possible that part of this effects here, all these protective and others, might be shared also by melatonin and on the way of HIAM and NAT, two enzymes which are responsible for converting tryptophan into melatonin and then finally into quinolinic acid, could be just of melatonin origin, which is present in plants also. So I mean in pasta. No one tested, I guess. But.
Yes, of course. Unfortunately, it wasn't tested, but we know that, for example, doing some researches, people writing projects measure the content of quinolinic acid properly. But of course, during preparing quinolinic acid in the quinolinic pathway, we have many substances, and it may be possible that they have positive impact on its function.
Any questions? So our next poster presentation is by Bugajska, and her topic is Evaluation of New Cryopreservation Methods on Human Amniotic Fluid-Derived Stem Cell Viability.
Thank you. Good afternoon, ladies and gentlemen. My name is Maria Bugajska, and I represent Nicholas Copernicus University. And I would like to present my electronic poster, which is entitled Evaluation of New Method Cryopreservation in 100% Serum on Human Amniotic Fluid-Derived Stem Cells Viability. Over the past few years, the huge attention is focused on stem cells, especially in the context clinical application. Fetal stem cells as an amniotic fluid is a novel source useful in regenerative medicine. And this kind of cells possess high proliferation and differentiation potential, and can be harvested without ethical reservation. As a standard method, the cells were frozen in DMSO, which is decrease the viability of cells. And this is why the new methods are needed. And the aim of my study is to analyze effect of cryopreservation method in 100% serum on viability of human amniotic fluid-derived stem cells. Materials and method. Samples of amniotic fluid were obtained from routine amniocentesis, and isolated cells were cultured in standard medium, supplemented with 20% fetal bovine serum, 10 nanograms per milliliter BFGF, and 1% antibiotic solution. We observed also expression of specific markers CD90, CD44, CD34, and CD45. And after reaching the homogeneous population of these cells and their appropriate number, after third passage, the cells were frozen with 100% serum and storage per fast 3 months. After the time, cells were thawed and viability was analyzed with the trypan-blue assay. And the results, we observed a very high expression of markers CD19 and CD44, which are characteristic for mesenchymal cells derived from different sources. And we did notice expression CD34 and CD45, which are homopoietic markers. Cells before freezing show fibroblastic-like morphology and the high viability rate. And however, after thawing, we observed the viability was reduced to 12.7%. However, despite this fact, cells were obtained, were cultured and seeded in the standard medium. And they did not show characteristic morphology. After 5 days of culture, all cells were eliminated. And this study showed that the cryopreservation method of with the use 100% serum is not appropriate for human amniotic fluid derived stem cells. The cells viability was reduced and the adhesion properties were poor. And as the conclusion that for sure that the further studies concerning the efficient storage method of amniotic fluid derived stem cells are needed. Thank you for much for attention.
Thank you, Bogańska. And if there are any questions by the jury?
This will be rather a comment because these cells were collected from amniocentesis, which is invasive procedure during diagnostic, prenatal diagnosis, exactly, yeah?
Yes, yes.
So it was a surplus material, probably, of karyotypic studies of the fetus, yeah?
Yes.
Right, and now tell me, please, what might be the advantage of these amniotic fluid cells, which are usually not very numerous, over conventionally collected umbilical vessels cells?
Could you repeat the last sentence?
Because it's quite common nowadays that couples after birth of a newborn are offered storage of stem cells from umbilical blood, actually from placenta and umbilical cord. And here you propose these amniotic fluid cells might be useful as a source for transplantation or regenerative active. What's the advantage of amniotic fluid cells over conventional newborn blood cells?
This is mesenchymal stems, so they cells can differentiate for another cells. For example, muscle cells for the regenerative or.
So you suggest these are pluripotent cells which can regenerate mesenchymal tissues. Okay, thank you.
Any questions? Thank you, Bogańska. I'll just pass you this certificate. Bogańska, thank you. So our next presenter is Daniel Adams, and he's going to be presenting the topic on microarray database mining reveals tumor rewiring of bioenergetic pathways, salvaging human glioblastoma mitochondria through substrate level phosphorylation. So I invite you to.
Sweet. Can I have a pointer? So hello, my name is Daniel Adams, and I represent the Department of Medical Biochemistry at Semmelweis University. And I'm here to discuss the metabolic changes which occur in glioblastoma cells to support their fastidious growth requirements. Glioblastoma is the most aggressive brain tumor in humans, as well as being the most common. Without treatment, survival time is around 3 months, and with treatment, around 1 to 2 years. Now, as you all know, there are 2 methods of ATP production within the cell. One is termed oxidative phosphorylation, in which energy is generated with the help of oxygen and the electron transport chain. However, it is well established in the literature that tumors live in hypoxic microenvironments not conducive to this form of energy generation. It is also established in the literature that the usual function of the ATP synthase enzyme, which usually generates ATP by the chemiosmotic potential difference between the transmembrane space and mitochondrial matrix, operates in a reverse mode in hypoxia. However, it was not known what the source of this ATP was to support this process, which brings me to my second method of ATP generation, termed substrate level phosphorylation. Today, I will be discussing the succinyl-CoA ligase G2 enzyme and G1, both subunits, within the mitochondrial matrix in substrate level phosphorylation. We determined a few years ago that substrate level phosphorylation was sufficient to maintain reverse operation of the ATP synthase enzyme within hypoxic conditions. And we also determined that glial cells, interestingly, lack this enzyme of succinyl-CoA ligase entirely. This enzyme right here. Other immunohistochemical studies also indicate a possible lack of alpha-ketoglutarate dehydrogenase, this complex right here, which precedes succinyl-CoA ligase. Glial cells are able to employ alternative shunt pathways around substrate level phosphorylation to convert alpha-ketoglutarate into succinate. So in the present study, we evaluated 5 different sources of microarray and RNA-seq data. Could you scroll up a little bit into this graph right here? So we evaluated 5 different sources of microarray and RNA-seq data to determine the expression levels of these enzymes supporting substrate level phosphorylation, namely these succinyl-CoA ligase G2 subunits, 2 subunits of alpha-ketoglutarate dehydrogenase, and the NME4 enzyme, which converts GTP to ATP, as well as the enzymes bypassing this in the alternative shunt pathway, namely succinic semialdehyde dehydrogenase, glutamate decarboxylase, glutamate dehydrogenase, and the OXY2-OXY1 enzyme, which converts, which basically bypasses substrate level phosphorylation right here. So what we found is that consistently the enzymes supporting substrate level phosphorylation were upregulated in every case in glioblastoma cells compared to healthy controls, both in microarray and RNA-seq studies. Also, the enzymes which bypass this pathway were consistently downregulated. These findings suggest that glioblastoma mitochondria do not rely on cytosolic ATP to maintain mitochondrial integrity. And since these enzymes that support substrate level phosphorylation are normally absent in glial tissue, we believe there's an exciting prospect for further chemotherapeutic research on a drug with minimal side effects. Thank you for listening.
If there are any questions by the jury.
I have one simple question. Is that shift in the source of energy for mitochondria inducible or permanent in the tissue? That means, can you mimic that by culture of these cells in low oxygen conditions? Sometimes these hypoxic conditions might be used for
Okay, so in hypoxic conditions, we found that no matter if it's healthy cells or tumor cells, that substrate level phosphorylation was sufficient to maintain the ATP synthase activity. We inhibited complex 1 as well as complex, so we inhibited complex 1, which abolished oxidative phosphorylation, and inhibiting, so I didn't exactly describe this because it's a little more complicated, but inhibiting complex 3, which prevents the regeneration of NAD in hypoxic conditions, converts mitochondria into ATP consumers. So even in healthy cells, substrate level phosphorylation is sufficient to maintain ATP synthase.
Okay, thank you.
Any questions? Thank you, Daniel. And here you go, please.
Thank you very much.
Now, the next presentation, I'd like to invite Dominique Tetera, and he's going to be presenting on the topic, measuring ethanol sensitivity in the diversity outbred mice population, implications for the indications of genes associated with alcohol use disorders in humans.
Oh. All right. Good afternoon, ladies and gentlemen, honorable jury. My name is Dominic Tetera. I'm a first-year medical student at Jagiellonian University right now, and I used to be a student at Millbury College last year, Millbury College in Vermont in the US. And since then, I've been working on a project on genetics of alcoholism. Alcohol use disorders are widely abandoned disorders with prevalence rates ranging from 0 to 16% of global adult population. A very strong risk factor for future alcoholism onset is the initial sensitivity to intoxicating effects of alcohol. And genetic contribution for ethanol sensitivity has been estimated to be approximately 50%. But finding specific genes that underlie sensitivity has proven very difficult. One of the major reasons for that is that majority of animal studies use crosses between inbred mouse strains with very few genetic recombinations. And these conventional approaches have only been able to locate a very broad chromosomal regions rather than specific genes. In this study, however, we are using a newly developed diversity outbred mouse population, in short DO, which displays a great deal of recombination. To model ethanol sensitivity, we used 3 paradigms. The first paradigm in ethanol-induced ataxia. So far, we tested 318 male DO mice on the latency to fall from accelerating rotarod. It's in here. Under ethanol-free conditions, and that was the day 1, which consisted of 10 training trials. And after intraperitoneal ethanol injection on day 2, which were the 3 testing trials on day 2. The second paradigm, which is ethanol-induced hypothermia, we tested 317 mice, and they received an IP ethanol injection, and the body temperature was measured at several time intervals. In the third paradigm, an ethanol-induced loss of writing reflex, 307 mice received an IP ethanol injection, and after losing the writing reflex, they were placed on their backs, and the time of the loss of this reflex was measured. On average, DO mice displayed robust responses to each aspect of ethanol sensitivity. The population showed a high level of variation in each phenotypic test, and the data are normally distributed, as can be seen on frequency distribution graphs here for hypothermia, or for a loss of writing reflex. There is also significant influence of ethanol on motor performance, which can be seen here, as well as the body temperature, which can be seen here in DO mice. There's also significant effect of learning over time on motor performance under ethanol-free conditions, which can be seen here, the progression and improvement over 10 training trials. We also showed that there is a slight positive correlation between body mass and loss of writing reflex, which can be seen here. And this very broad range of responses means that the population has high variation in ethanol sensitivity phenotypes, and phenotypic diversity is likely correlated with genetic diversity. Ultimately, we plan to test a total of 576 mice, densely genotype them using the MegaMuga genotyping array, and perform quantitative trait locus mapping, QTL mapping. Only highly recombinant and genetically diverse population of mice used in this study will provide a much better resolution for QTL mapping and potentially enable us to identify candidate genes that underlie ethanol alcohol sensitivity. Moreover, we'll perform RNA sequencing in order to look at differences in gene expression that are associated with behavior and genotype. By linking genetic variation and expression to neuronal function and behavior in mice, we predict it may be possible to target specific molecules to prevent and treat alcoholism in humans. Thank you.
Any questions from the jury?
I think we still have that. Yeah.
Just thank you very much. Well, the route of administration of ethanol, am I right? I've heard it was just by injection, right?
Yeah, intraperitoneal injection.
Okay, but that's unusual comparing to humans, perhaps.
Yeah,
right?
that's true.
But of course, the target of the studies were different, right? So, and I'm not sure whether this study went so far, but perhaps any metabolite of alcohol were tested or were studied, like aldehyde. It is converted into aldehyde, perhaps.
Oh, not yet. No. Yeah.
Okay, thank you.
My more general question is that, in fact, you are looking at the tolerance of ethanol using this evaluation of its effects following administration. Do you expect also to find a correlation between this quantitative trait loci and addiction, which are usually tested using different psychosomatic tests of place preference or whatever? This is one question. And the other, do you expect that this sort of finding genetic determinants to alcohol tolerance can explain another problem with its overuse, which is fetal alcohol syndrome? Does it exist in mice? Can be reproduced? Abnormality in fetal development induced by ethanol.
All right. So answering your first question, we are hoping to find candidate genes that might play a role in ethanol sensitivity. So hopefully, and since the ethanol sensitivity is genetically a risk factor for alcohol dependence or alcoholism, we hope then at first find those genes, and if we do find them, then we probably will try to test those genes with different protocols. Probably more for addiction rather than sensitivity. Yeah.
And what about FAS, fetal alcohol syndrome?
That's a very good question. I'm sorry, I do not know the answer to it. Sorry.
Thank you.
Any questions? So thank you, Dominic. I'll give you this certificate now. Our next presenter is Katarzyna Kopasz, and I'd like to invite you.
Ladies and gentlemen, my name is Katarzyna Kopasz and I represent the Medical Society of Scientific Society of Medical Biochemistry, Jagiellonian University. The scientific paper I would like to present is called The Use of Titan Yellow as a Marker, as a metal ions binding marker for studies on formation of complexes by supramolecular Congoret. Congoret is a supramolecular ligand because interact with proteins as an assemble of molecules. Congoret can also interact with amyloids or carbon nanotubes. It can be used as a contrast agent in electron microscopy studies to analyze protein structure, providing they will be labeled by elements like silver ions. Yet, they are not create stable complexes, so the indirect method had to be developed. The aim of this study is to verify if Congoret and silver can combine titan yellow and create stable complexes, which can be used like as a contrasting agent in electron microscopy studies to analyze protein structure, amyloids in particular. Concerning the results, the significant changes in absorption spectrum in spectrophotometrical analysis and changes in speed of mobility in electrophoresis separation proves that Congoret, titan yellow, and silver create stable complexes which can be used as a contrasting agent in electron microscopy studies. During the surgery occurs that also titan yellow can bound amyloids alone. He doesn't need Congoret to this. It's interesting because we can analyze structure of these proteins. What's more, these complexes can also interact with carbon nanotubes. In the picture, arrows indicate complexes combined to the surface of carbon nanotubes. It is worth meaning that carbon nanotubes are currently studied as potential drug carriers. And conclusion, understanding the molecular mechanism of amyloids are very important in therapy and diagnosis of amyloid disease. Thanks to the combination of amyloids and Congoret with titan yellow and silver complexes, a new method of contrasting agent in electron microscopy studies is proposed. The preliminary results presented here require future studies. Thank you.
Are there any questions by the jury? Okay, there are no questions, and Katarzyna was the last presentation for today, and We'd like my colleague Kinga, she will present the results shortly.
Yes, I think that we should have the results in 15 minutes. So at 7 p.m. you could be here and wait for it. But the official certificates and prizes will be actually given to you on the closing ceremony. Today, just the announcement of the results.